Good morning, students. Good morning. He has adjusted now. Audio. Is the audio all right now? Thank you so much. Thank you so very much. Lots of blessings for this day. And the whole week, you should remain blessed day by day with the utmost best of your health, best of your mindset, best of your energies, so that on 12th, you will perform the best of your life. The best of your life. So the whole week should be very much confident. Never ever the confidence should go down. Yes, fluctuations do occur, but it should not be a continuous one. So this marathon class, whatever you call it, it's a long class, and we'll try to do the main points. In the revision classes we have done chapter by chapter, but here it is not chapter by chapter, it is the whole zoology, the whole 50 questions, wherein you have to write 45 questions, 45 answers you have to write. Okay, so I start with main points where the students make mistake, and those are the favorite points for the teachers to make questions. For example, I'll start with this animal kingdom, the chapter number 1 in zoology, class 11, first year chapter. Fertilization, fertilization in different phylum, and these are the names of the phyla. PO for porifera, CN for nidaria, like that. Okay. And just a sec, I'll just have a little water. Thank you. So, my dear children, till HE is hemichordata. So, you can see this hemichordata. Now, what you can do, you can remember that in all these non-chordata, the internal fertilization is in PPAA, and that stands for porifera, platyhelminthes. Then we have the eschelminthes, and then we have Annelida and we have Arthropoda. I'm sorry, not eschelminthes, it is Annelida and Arthropoda. I repeat, porifera, platyhelminthes, these two P and two A's means one is for Annelida and one is for the Arthropoda. Usually in Arthropoda, fertilization is internal. However, in some aquatic form, fertilization may be external, but in general, it is internal. So please remember this thing. Now, chordata may have both external as well as internal fertilization. Now, there are also some specificities there, but for the examination point of view, just remember this. Chondrichthyes, the cartilaginous fish, they have internal fertilization, and all the amniotes, they also have internal fertilization. Amniotes. Those who have amnion in their development, amnion is an extraembryonic membrane. We know that there are 4 extraembryonic membranes. One of them is amnion. You might have heard about amniotic fluid. It's related to the same amnion and amniotes. Okay, so 3 of the classes, these are class of phylum chordata, and remember the name RAM, that is RAM, RAM, Reptilia, Aves and Mammalia. And in these 3 class of chordata, there is internal fertilization. Okay, usually they ask you from this only. They won't be giving you very specific, because in animal kingdom, okay, so we may have, like for example, you may find someone in Annelida where external fertilization is there, right? Okay, someone in Annelida like that. So then there is another thing, is cloaca. Cloaca, it is a common chamber wherein the digestive tract and the reproductive tract and urinary tract open up. Please remember, it is mainly found in the vertebrates, cloaca in the vertebrates. So don't try to search for it. Yes, we do give, in some roundworms, we do give a name cloaca, but generally the word cloaca refers to the vertebrates. And yes, in our embryo also, there is a cloaca, okay? And so cloaca is found in the chondrichthyes, then amphibians, reptilia, and birds. Again, are amphibia, reptilia, and birds, where you have cloaca. Okay, of course, in mammals also, some mammals have cloaca. For example, the egg-laying mammals or even the marsupials, they have cloaca. I don't think so. Only this much you should remember what they have given in the NCERT. My dear children, I want to correct myself. To start with, this trick that we have made, okay, the trick that we have made, which is PAPA, actually P stands for the Porifera, perfect. And now these two are the worms. I mistakenly said Annelida. It is Platyhelminthes, then this is Aschelminthes, and this is Arthropoda. Platyhelminthes, Aschelminthes, and it is Arthropoda. How to remember? Please keep it in mind that these two are worms. Okay? Like that, you can keep it in mind. And how to remember that it is not Annelida. Annelida, for example, in earthworm, the main annelid, in earthworm, the fertilization is in the cocoon, outside the body and inside the cocoon. Okay? Yes, there are some annelids where there is fertilization inside the body, for example, in Nereis, but then in general, what we have to answer is external fertilization. This is one question which you should be knowing, okay? And in this chapter, many questions can be framed from levels of organization, like tissue level. One more fact you can note down here, and which is also mentioned here, that all diploblastic animals have radial symmetry and are acoelomates. Please note down, all diploblastic animals have radial symmetry and are acoelomates. Okay, so one more gist that you can find out here. And again, I want to say again, emphasize again, Porifera, then these two are the worms like flatworms and the roundworms, and this is Arthropoda. Arthropoda, usually it is internal fertilization, okay? And internal fertilization in the chordates also, in chondrichthyes, in the cartilaginous fish, and in the reptiles, amphibia, reptilia, and mammalia. Okay. Circulatory system, open type and closed type of circulatory system is again a place where we make questions and ask you which of the following is also having open type of circulation. It is diploblastic, it is triploblastic, like that. So examples of open and closed type of circulatory system one should keep in mind. Now closed type of circulatory system, remember that chordates have one of the like subphylum wherein we have urochordate. Urochordates have open circulatory system, but still this is not given in the NCERT. So chordates we will consider as closed circulatory system. Chordates closed circulatory system. Chordates closed circulatory system. Please keep this point in mind. Okay, Arthropoda closed circulatory system, Arthropoda open circulatory system, Annelida closed circulatory system. These points one should keep in mind. Symmetry, you can easily understand symmetry. Yes, bilateral symmetry is an advanced feature than the radial symmetry. Bilateral symmetry is an advanced feature. Echinodermata, the adult may be pentamerous, radially symmetrical, but their larva shows bilateral symmetry. So, and even in case of the gastropoda, mollusca, in mollusca gastropoda, adults are asymmetric. However, their larva is bilaterally symmetrical. So that way, please remember, please keep it in mind that bilateral symmetry is an advanced feature. So my dear children, one more work you have to do here is just go through the examples, all these examples, name of these examples. I think it will take another 15-20 minutes of yours to do this, and do this 2 to 3 times, because if you are confident about the example, I think more than 50% confidence you will have about writing the answers. Yes, they will not be asking you very, very detailed and very, very detailed exceptions which are not given in the NCERT. Then we have the next chapter, structural organization in animals. Okay. Now this chapter has got various parts like animal tissues. Then we have the 3 invertebrates. Just 1 second. क्या हुआ? मुझे शाहजहां का नंबर सेंड करना. क्लास में नहीं है. बाबू से लो. बाबू का है. Okay. So structural organization in animals. Okay, and it has got animal tissues. One question certainly from the animal tissues that will be asked. Now about those non-chordate animals, 3 invertebrate animals are given there. 2 invertebrates and 1 vertebrate is given there. Like we have earthworm, we have the cockroach, and a vertebrate, a representative vertebrate frog is given. There are many confusions that it is not a part of the syllabus which they have mentioned. However, because animal kingdom is in the syllabus, if they give a detailed question on any of this, the phyla or any of the class of chordata, we cannot stop them. There is nothing like we can legally say to them. Anything in the NCERT book is the syllabus. Moreover, people are also preparing the previous NCERT books, the older versions also. However, it is not that important, but at least whatever given in this NCERT book, that has to be done completely. Okay. No, please don't take any chance. Okay. The maximum questions can be asked. I hope I have shared with you all the distribution of the questions, the distribution of the questions. If I have this, I'll show you here itself the distribution of the questions in different topics of zoology. Okay. If I have that here. Actually, this is a different icon. A different icon is coming. Distribution, chapter-wise weightage, neat zoology, chapter-wise weightage. So, what we have seen from 2016, and in some of these years like 2016 itself, there were 2 papers. Even 2020 also, there were 2 papers, like one was the main one and one was Odisha one. So please keep all these. We have what we found that animal kingdom 3 to 4 questions. 4 is also on the higher side. 3 questions, what I can think of. Then structural organization in animals, like 2 questions, not even 3, 2 questions I have seen which can be asked. Sometimes they drop it, but then 2 questions because there are various parts which should be kept in mind. Majority of the questions come from human physiology. That means you can see, now these are not the marks, these are the number of questions. Okay, so from every chapter, minimum if you take 2 questions from each and every chapter of the human physiology, because that is very important. Biology and human welfare, from this unit we have 3 to 4 questions from human health and disease, and 1 question from strategies for enhancement in food production, animal husbandry. Then applications of biotechnology in medicine, 1 question, because it has got different applications in agriculture also. So chances of a question coming from medicine wala point, medicine wala part is 1. Ecology, now this is something important, ecology, the next higher, number of questions after human physiology is from ecology chapters. Organism and population, ecosystem. So what we find that more or less 2 to 3 questions fluctuating in these chapters, 4 chapters of the ecology. In ecosystem, only the zoology portion, because some portions that comes in the botany of our distribution that we do here. After that, we have genetics and evolution, both genetics and evolution, 2 chapters of genetics. However, we have only a last part of the chapter, not the complete chapter, because from genetics also come in botany and also comes in zoology. So more or less to be on the higher side, this number, you can say evolution 3 questions, molecular basis and principles of inheritance 1 to 2 questions, okay? And reproduction is both reproduction and reproductive health, 5 questions. So I think I have shared this with the online one, might be some of you have missed it, and the offline one, I am sharing it with you right now. So, and this is what you see the result, the scorecard, and this is the place where you write the number, and you always visualize that you are writing your number and you are getting a very good message from that. Create your result in advance with the power of visualization. So never visualize yourself being trapped by anything, being not scoring well in the examination. Yes, you may see some dreams, don't worry about that. But in conscious mind, never focus on anything, the result that you don't want. If you want that result, it's fine. But if you want a good result, it is better to focus in the good result only. So keeping this perspective in mind, the human physiology has got maximum questions. And in the human physiology, in every chapter, there are certain places where questions can be asked. For example, the enzymes in case of human digestive system, digestion and absorption. I think I have that here, digestion and absorption. Let me just check whether we have that or not here. Digestion and absorption. Only the part which is given in the NCERT, the enzymes, and what do they do after digestion, what do they do. Even in today's question also, we have given some questions based on that. So this part, one should make it very clear because that is an important part. Type of teeth, though we all know the type of teeth in human beings, not a big deal. Dental formula, we have done it very, very nicely, very, very detailed. We have done it and not a big deal. Now, here what you see, the number of roots that you see here. Of course, these numbers are not the number of roots written. No, these are not the number of roots written here, okay? But the number of roots in the teeth, in incisors and canine, and also in case of premolar. There are single root except the upper maxillary, means upper premolar, first premolar. Otherwise, mainly in the molar, either there are 3 roots or there are 2 roots. In the upper jaw, molar 3 roots, and in the lower jaw, molar 2 roots. Upper 3, lower 2, upper 3, lower 2. Upper jaw molars 3 root and lower jaw molars 2 roots. Keep that point in mind. This again a diagram which I have seen in many of the questions given by different institutes, also different coaching centers also, that this is an important diagram. Directly diagram can be given, so the sequence of the layers from outside to inside, serosa, then muscularis, after that submucosa, and then mucosa. From inside to outside, mucosa, then submucosa, muscularis, and serosa. In the muscularis, remember that these are smooth muscles, number 1. And number 2, there are 2 types of muscles, longitudinal and circular. Here you should remember, all is outer longitudinal, inner circular. outer longitudinal, inner circular. OLIC, OLIS, please remember this point in mind. Never ever forget the role of these lacteals in absorption of fats. Lacteals are important for absorption of fats. Now here comes your different digestive juices and just a gist of all the digestive juices that we have and the glands that we have. The digestive glands, just 1 second, let the board opens up. Okay. So we have the digestive glands like salivary gland, then we have salivary gland, then we have the gastric glands, liver, pancreas, and small intestine. I repeat, small intestine, pancreas, liver, gastric glands, and salivary glands. And they produce different juices. For example, the salivary glands, they produce saliva, a juice called saliva. Yes, it is considerable. You know, the amount of saliva produced in one day is equal to the amount of gastric juice produced in one day. So good amount of saliva we can expect. It evaporates, therefore we are not knowing that. So it is an important digestive juice. Then we have the gastric juice. After that, liver produces bile. Remember that gallbladder is only storing the bile, but liver produces bile. And then we have the pancreatic juice. And lastly, we have Circus entericus. An idea about the pH of different juices. The pH of different juices, for example, the pH of saliva is 6.8, of gastric juice is 1.8. Okay, it's given in the NCERT is 1.8, better you remember 1.8. Bile is 7.8, and also Circus entericus is also 7.8. So bile and Circus entericus is 7.8 each, and pancreatic juice is having the highest pH of more than 8, which is 8.4. So in all, 8 is the following number, but only in the pancreatic juice 8 comes first and then 4. 8.4. Maximum pancreatic juice and minimum pH is of the gastric juice. So this is important because all the enzymes present in that juice have their pH optima as the pH of that juice. Now, saliva has got enzymes. For example, there is an enzyme called salivary amylase. Then we have the lysozymes. Of course, they don't have any digestive function, but they digest the microorganisms. So lysozymes help in digesting the cell wall of bacteria and act like an antibacterial. Salivary amylase, as you know, it is a starch digesting enzyme. Can I expect amylase to digest glycogen? Yes, why not? Glycogen and starch both can be digested by salivary amylase, okay? Gastric juice, the enzymes present in the gastric juice, we have pepsin. Now, there is one doubt that can come to the mind. Sir, in stomach produces pepsinogen. So, if it is written pepsin, shall we consider it right or wrong? Correct. So, see, if someone asks you the source of pepsin, of course, we will say that this is stomach. If they particularly ask you this, that the stomach produces this enzyme in the active form, then that is a wrong statement. Otherwise, it is a generic statement. It is a generic statement. For our understanding, we should know that the protein digesting enzymes are produced in their inactive form. Okay, so we have pepsin, then we have another enzyme called renin, R-E-N-N-I-N, renin. Of course, renin is also produced in its inactive form, prorenin. Bile does not have any enzyme. Pancreatic juice is a complete digestive juice. It's a complete package, complete digestive juice. It has got enzyme for all the components, and remember that once the food comes in the small intestine, the first one to act on food is pancreatic juice, and tarvata is Circus entericus. The Circus entericus is the last one to act on the semi-digested food. Semi-digested food. Last one to act is Circus entericus. That means Circus entericus is giving the final touch. The final touch in digestion is given by Circus entericus. When the food comes, when the chyme comes into your duodenum, the first juice to act upon it is pancreatic juice. Pancreatic juice has enzymes. For example, it has got nucleases. It has got nucleases, ribonuclease, deoxyribonucleases. Okay, so we can call them DNAase and RNAase. These are the nucleases. Okay, then there are protein digesting enzymes. What are those in the pancreatic juice? We have trypsin. Of course, trypsin comes as trypsinogen. Okay, but trypsin or trypsinogen. Okay, I'll write trypsinogen. Then both chymotrypsin or chymotrypsinogen, and one more is procarboxy peptidase. Procarboxypeptidase. So these are the protein digesting enzymes. These three are the main protein digesting enzymes. Then we have the amylase, pancreatic amylase, the important amylase. Yes, saliva has amylase, but in the buccal cavity, the starch is not digested completely. Max to max starch up to 30%. How much? Up to 30%. Max to max 30% can be digested by salivary amylase, and rest of it is digested by the pancreatic amylase. In the stomach, there is no amylase, so there is no digestion of the starch. Then fats, and the fats digesting enzyme is lipase. Now you can see that this lipase is the most important lipase in the human digestive system. I repeat, pancreatic lipase is the most important lipase in the human digestive system. However, we have another lipase present in Circus entericus, but its amount is very, very less, very, very minimum. So if the pancreatic lipase is not secreted, there will not be any digestion of fats, and it will lead to fatty diarrhea called steatorrhea. I mean to say, please keep it in mind that pancreatic lipase is an important lipase. Now, what you saw here, that there are nucleases. These are the nucleases, DNA, RNAs, protein digesting enzymes, carbohydrate digesting enzymes, and lipids, fats digesting enzymes. Another question, which is the main fat present in the human diet? Answer, triglycerides. Now, another question, what are the enzymes present in Circus entericus? In the Circus entericus, nuclease present in pancreatic juice. So here we will have nucleotidase and nucleosidase. Complete nucleotidase, tarvata nucleosidase. Okay, and then finally we will get the nitrogen bases and sugar. Complete digestion first started here and then completed here. Protein digesting enzymes, we have dipeptidase. dipeptidase, and this is not in inactive form, dipeptidase. So this is active form. It will digest the dipeptides into 2 amino acids. Dipeptidase digest dipeptides into 2 amino acids. Okay. Now coming to the carbohydrates. Carbohydrate digesting enzymes are disaccharidase. What did I say? Disaccharidase. I mean to say, please keep this point in mind, my dear children, that there is no amylase to be expected here. There is no amylase you can expect here. So I'll write here amylase, and I will cross amylase here so that you can keep this point in mind, that there is no amylase in Circus entericus, and that is a question. So what are the different carbohydrate digesting enzymes present in Circus entericus? The different carbohydrate digesting enzymes are disaccharidase. For example, we have got maltase, we have got sucrase, we have got lactase. So these are the disaccharidase. Maltase digests maltose into 2 molecules of glucose. Sucrase digests sucrose. Sucrose is the table sugar, panchadara, the chini, the table sugar. Sucrose into 1 molecule of glucose and 1 molecule of fructose. Sucrose into glucose and fructose. Maltose into 2 glucose. And lactose into 1 glucose and 1 galactose. 1 glucose and 1 galactose. Ante, glucose is common in all 3. Galactose is only present in animal, in the, it is the only sugar of animal origin, galactose. Okay, guys? And a very small concentration of lipase can be seen here. However, it is not an important one, but lipase, intestinal lipase is present here. So this way you can recall, this is for nucleic acids, final digestion. This is to complete the digestion of peptides, the proteins. This is to complete the sugar digestion, the carbohydrates. And this is to complete the fat digestion step by step, triglycerides into diglyceride, first by pancreatic lipase. After that, diglyceride into monoglyceride by pancreatic lipase. And now monoglyceride to glycerol and a fatty acid, the last fatty acid, glycerol and fatty acid by the Circus entericus lipase. So Circus entericus lipase is just acting on monoglyceride, and pancreatic lipase acting on tri and diglycerides. So my dear children, this is an important thing, and this can make you understand that which enzyme is present where. Mainly the problem comes in these two. There is no such enzyme here. This we can just remember that potalopepsin and renin, and this is conditions apply that only in case of children, infants and children, and the name itself is salivary amylase. So obviously salivary amylase will be in saliva, and the lysozymes are the antimicrobial enzyme. Lysozymes are produced by Paneth cell in crypts of Lieberkühn. My dear children, one more point. Saliva by 3 pairs of salivary glands. Yes, they are in pairs, 3 pairs of salivary glands. Gastric juice by the gastric mucosa. Now, bile by the liver cells. Pancreatic juice by pancreatic acini. Pancreatic acini means the exocrine part of pancreas. Pancreatic acini. also called pancreatic lobule, pancreatic acini, succus entericus by crypts of Lieberkuhn, crypts of Lieberkuhn, Lieberkuhn or Kuhn, Kuhn, right? Crypts of Lieberkuhn are the glands present in present in small intestine. In the large intestine, some mucous glands are present, but there is no such gland which produce any digestive enzyme. That means all digestion gets over in the small intestine, my dear children. Hope it is clear to everyone. Now the steps of digestion one has to just go through. Now if you see the steps of digestion, and once you know that which enzyme is in which compartment, it won't be difficult for you. So you have to just see that what it does. It is quite obvious that amylase will work on the starch. And if starch is acted upon by amylase, in the first step, it is not going to do any complete digestion. It won't complete the digestion of the sugar, the starch. So first, what it will form? Maltose, isomaltose, and then maltose and isomaltose means it's an isomer. Okay, just name of the bond. The bond is different there, and both are almost same, and both yield glucose. Both of them yield glucose. So please keep all these points in mind, my dear children. This is about the digestion. I don't think so. And similarly in proteins also, you will not get directly, you will not get the amino acids. Any complex protein acted upon by pepsin will give you proteoses, peptones. What are these? These are the broken pieces of the proteins. Okay. Above all, last 2 to 3 years, we have seen a question has been asked. Above all, an important enzyme here, and I call it an activator enzyme. It's an activator enzyme. It is not, this enzyme is not digesting any food, but it is just cleaving a part of the peptide bond and making one protein digesting enzyme active. And this activator enzyme is activating one protein digesting enzyme, and that is trypsin, in the inactive form trypsinogen. Can you please name which enzyme converts trypsinogen into its active form trypsin? Let me tell you, let me help you a little bit that there are two. I mean to say, of course, that enzyme is there, and along with that, once this trypsin is active, that trypsin can also activate trypsinogen. Once trypsin is active, it can also activate trypsinogen. Please tell me which enzyme is doing this, and is it at acidic pH or basic pH? Now the questions should include, the answer should include the name of the enzyme and also the source of the enzyme. Source, come on. So the classroom, so we cannot have answers here, but the online children, we can have answers here. Come on. Small intestine. Well, I take that. Source is small intestine. I take that. Small intestine glands are crypts between the villi, and the enzyme. Enzyme is enterokinase from Circus entericus. Complete answer. Enterokinase. My dear children, on enterokinase, questions have been asked in last 2 to 3 years papers. If you check, enterokinase and Bruner's gland are important. 2017 to 2020, 2 to 3 times, even this year also, 2020, if you see the paper, enterokinase was in the question paper. Already we have told you, because 2017 onwards, this question is coming. Okay. Please remember that the source is small intestine, crypts of Lieberkuhn, and it is acting on the enzyme of pancreatic juice. So this is coming from a different juice and activating the enzyme of a different juice, okay, in the alkaline pH, This is an important point and should never be forget. Another thing is another gland called Bruner's gland. Bruner's gland. Now this gland is present in the submucosa. This submucosa is very important to note here. Submucosa of dash, and the enzyme produced by this. Come on, and the enzyme produced by this submucosa of dash. This submucosa is important. I'll be telling you. I'll put some star mark here, asterisk sign. Come on. Bruner's gland, they are present in the duodenum. Very good answer. They are present in the duodenum. I think 25 answers I got. Yes, it is duodenum. Good. And enzyme produced by this. Bruner's gland. It's only 2 computer, 1 TV. We are not able to check the answers of other one. Anyways, yeah. Anyways, I'll try to switch over this to this. So right now I'm seeing one of the TV. So enzyme produced by Bruner's gland. I'll give the answer myself. There is no enzyme. No enzyme. It is not producing any enzyme. It is producing mucus and some sodium bicarbonate and a alkaline mucus. Bruner's gland, submucosa of duodenum. Please remember, this is special. This Bruner's gland is special, my dear children, because all the other glands are in the mucosa. All the other glands are in the mucosa. For example, crypt of Lieberkühn is the extension of mucosa. For example, gastric glands, extension of mucosa. But the submucosa only in the duodenum. Again, this is a special point wherein teachers can give you a question, and this is every student almost these days are doing. We have to see the answers which everyone does. We have to make them correct, and also the one which others don't do correct and we will do correct, then only we will be in the merit list. Okay, my dear children. So this is all we can discuss at this. I'm not saying these are the only points to remember in digestive system, but then this is the important part. Diagrams of this chapter, you can see these are the diagrams. Okay, liver is not shown here, but this is a C-shaped duodenum is shown, and in the concavity, this gland which is seen is a pancreas. And what opens in the duodenum is a hepatopancreatic duct. Hepatopancreatic duct is formed by the merger of common bile duct and pancreatic duct. Both of them join to form hepatopancreatic duct. And hepatopancreatic duct, at the opening into the duodenum, there is a sphincter in the hepatopancreatic duct. Anyone can tell me the name of the sphincter, guys? Anyone, please revise this name of the sphincter. Okay, thank you so much. Thank you so much for wishing me Teachers' Day. Thank you so very much. Yes, Audi, very, very good. The sphincter of Audi, very, very good. This is a very good wish that you are doing, answering so fast. So really, the teacher feels very happy, very contented, and let me tell you, very, very proud by seeing such quick answers. Okay, because there are not, some special questions. Those generic questions are there, which can be expected here. Food remains in the stomach for 4 to 5 hours. Food remains in the stomach for 4 to 5 hours. Please understand this. Please keep this point in mind. Okay. Once you have the name of the enzymes and you know what outcome it comes, then you can just understand. There is no need to batti kotu. There is no need to, mug up all this. But what you have to understand that where is this dipeptidase, where is maltase, lactase, sucrase, nucleotidase, nucleosidase, lipase, all these are in Circus, Entericus, like that. So this we have to remember, we have to understand. Calorific value, these are simple points, like you have to just remember. There are two. One is the calorific value of food and one is a physiological value of food. Calorific value of food is in bomb calorimeter, so we have more accurate values like 4.1 kilocalories for carbohydrate, 5.65 and 9.45. 5.65, 9.45. 5.65 for protein and 9.45 of fats, which I have in my body plenty. But when we eat them in our system, in our calorimeter, in our body, it just gives 4 kilocalories per gram carbohydrate, 4 kilocalories per gram of proteins, and 9 kilocalories per gram of fats. That is maximum our body can obtain from these three different types of food. Absorption of food is again an important thing. Absorption of food, there is a lot of confusion given here. Even the NCERT guys also know that there is a confusion, and once the mistakes done earlier, then it is difficult to directly rectify them. But what you have to answer, what questions can be given. For example, they can ask you for anything. They can ask you for glucose. They can ask you for amino acids. And they will ask you that how they are absorbed. They are absorbed by, if they give you by simple diffusion, answer is correct. They are absorbed by simple diffusion. Okay. However, this quantity is very, very less, only till there is a gradient, some amount of glucose, some amount of amino acids, and even some amount of sodium ions, electrolytes can simply diffuse inside the epithelium, inside the membrane. So simple diffusion, but only small amount. Remember, it is a small amount. Then majority of it is by facilitated transport. So facilitated transport, that is the majority one. And is it dependent on energy? Is it dependent on energy? Yes, it is dependent on energy. The absorption, majority of the sugar, majority of the glucose and amino acids, they're absorbed actively. So we can also call it an active transport. So please remember these three, all three. So if a question comes, the answer will be all of the above. As seen the last year trend, my personal assumption is the paper will not be any different than what you have seen in the last year. Any different than what you have seen in the last year. In the last year, that means in 2020, the match the column were very easy, because if you can match any one of them, even if you don't know the other, you can get the answer. That type of ease might, because then what's the use of match the column, right? So that type of ease might not be there this year. And don't worry about that. If you find the question paper difficult, remember that it is not only for you, it is a paper given to everyone out there, and everyone will be facing the same problem. Remember that there is no such target we have to hit here. Yes, our target is maximum, but if the paper is difficult, then obviously that maximum will go down and the cutoff will also go down. In the last 4 or 5 years, there has been evolution from where the student get very easily, they can get the seat in general category in 500 marks to more than 600 marks. This evolution has happened. Okay, so don't worry if the paper is difficult, then again, this cutoff will go down. Hope guys, you can understand this. So you should not be worried about the type of paper that comes to you. Now in this one, in the summary of absorption in different parts of the digestive system, remember that D and D are given in first and the last part of the alimentary canal. That means drugs are absorbed in the buccal cavity. You know that some drugs can be kept underneath the tongue and they can be absorbed in the capillaries underneath the tongue, and some drugs are absorbed in the large intestine. In the small intestine, because of the acid, some drugs are not able to open up, or some drugs are not able to work properly. So they are being coated in such a way that they will only open when they are in the large intestine, and there they they will be absorbed. Okay, and remember that in the small intestine, everything is absorbed in the small intestine, everything. So rather than small intestine, if we can memorize these three, then we can say rest all is in the small intestine. And needless to say again that fats, the digested form of fats are fatty acids and glycerol. They are absorbed in the lymph. Okay, guys, and the disorders. In the disorders, needless to go to very detail. Just remember, guys, jaundice. This, remember, it is bile pigments and not bile salts. Bile salts are important thing. They are not the waste products. Bile pigments are the waste products, my dear children. And because the liver makes bile pigments and secrete the bile pigments in the bile juice, that is the reason we consider liver as an accessory digestive organ. Accessory digestive organ. Okay, guys, so please, the reasons for indigestions are plenty of reasons. For example, inadequate enzyme secretion, anxiety, food poisoning, overeating, and if you eat spicy food, then also the digestion is affected. Digestion is affected. Protein energy malnutrition, particularly India, condition of India is much improved from the years when we were making our studies and from the years where you are studying. So conditions are quite improved, but still there are many parts in India also where there is severe malnutrition of the growing or the young kids, young children, young babies. So please remember that kwashiorkor is a type of protein energy malnutrition where proteins are mainly deficient. These guys are getting, because you know that in some in some governments, in some countries, the government provide rice, staple rice for the poor people. And boiling the rice, mixing some uppu into it and eating the rice is not going to give all the nutrition. In the adults, it might be okay, but for the young children where the proteins are really required, they are really suffering. So yes, we call it a PEM, protein energy malnutrition, but in P and E, it is mainly P component which is deficient in case of kwashiorkor. So please remember K, kwashiorkor, with P, protein. And if proteins are deficient in the body, if the blood proteins are low, can I expect the blood colloidal osmotic pressure to be normal? No, because blood colloidal osmotic pressure is also related to the amount of proteins in the plasma. Can you please tell me which protein? We have two types of proteins now. Some proteins come in the globulin category and some proteins come in the albumin category. Come on. Which protein is responsible for blood colloidal osmotic pressure? Come on. It is albumin, correct. Albumin. So my dear children, if proteins are deficient in the body, the blood colloidal osmotic pressure will be low, and hence the blood is not able to retain the water into it. That water comes out and then deposit in the tissues. As a result of that, you will find swelling because water has come out now. You will find swelling in these children. So they have swelling, and the medical term for swelling is edema. O-E-D-E-M-A, edema, the medical term for swelling. So swelling is there in kwashiorkor, and complete emaciation is there in the other disease, which is a complete protein energy malnutrition, PEM, complete protein energy malnutrition. And the worst part is this complete protein energy malnutrition, which is called marasmus. It affects children younger than 1 year of age. If 0 to 1 year of age is called infancy, infant. The age group is called infant, 0 to 1 year. Less than 1 year, we have marasmus. And the child will totally emaciated. I use the word emaciation, emaciated. Even the examiner can use this word. It means extreme weakness, complete thinning of the body, wrinkling of the skin will be seen. So in one, you will have pot-bellied, like me. In one, there is complete thinning, complete thinning. So marasmus, there will be complete thinning, and kwashiorkor, there will be little puffiness that can be seen. So if you can recall all this, I don't think so, you will be having any trouble in answering these. Coming to these, whenever you are doing, just go through these questions, just the objective type of questions. Objective means where the answer is a word or just true and false, that is objective. The other answer where you have to describe something is called a subjective answer. Okay. Now, in case of the respiratory system, some places where students still have doubt is the mechanism of breathing. In the mechanism of breathing, one thing you have to remember, that if the volume of the thoracic cavity increases, the volume of the lungs will also increase. And applying the Boyle's law, the pressure inside the lungs will decrease. And the moment pressure comes down lower than the atmospheric pressure, the air can be sucked inside. Remember that because of the diaphragm, the type of pulmonary breathing in case of mammals is quite different from the pulmonary breathing in other vertebrates. Can you please tell me what are the vertebrates which breathe through pulmona, which breathe through lungs? Name the vertebrates. If for reptile you write R, I will take that, no problem. Please say what are the vertebrates which have lungs, which breathe by lungs. Vertebrates. Mollusca is not a vertebrate, beta. And molluscan lungs, the name is given lungs, but they are ctenidia, a special type of gills called ctenidia. Yes, we have got amphibians, reptiles, birds, mammals. Amphibians, reptiles, birds, mammals all have lungs, pulmonary breathing. The pulmonary breathing of mammals is quite different because mammals can create a negative pressure inside the lungs. And for that, they have something by the name D, and that D stands for diaphragm. Okay, guys, so when the pressure inside the lungs decrease, air is sucked inside, and vice versa, when the pressure inside the lungs increase, the air exhaled out. Okay, please remember that what are the things that can increase the volume of the lungs and decrease the pressure. So how the pressure can be decreased, how the pressure inside the lungs can be decreased, and of course, how the pressure can be increased, okay? I'll start with the decreased one. And what is the significance of the decreased pressure? The significance is that when pressure inside the lungs decrease, air will go inside, inspiration takes place. My dear children, pressure will inside the lungs decrease by contraction of external intercostal muscle, EICM, external intercostal muscle. Then the diaphragm muscles, phrenic muscles, or simply you can call it diaphragm. Diaphragm, contraction of diaphragm will also cause decrease in the pressure inside the lungs. So external intercostal muscles and the diaphragm, they decrease the pressure inside the lungs. How the pressure inside the lungs can be increased? The pressure inside the lungs can be increased by simply relaxation, of these two. What are these two? EICM, external intercostal muscle, and the other one is diaphragm. Hope you can recall, you can understand this by relaxation. And also, can you have any other muscle which contraction causes increase in the pressure inside the lungs? Now I'll be seeing the other TV to get the answer, because there are two TVs, two laptops here. Yes, guys, can you please tell me? Guys, please tell me. My question is, any other muscle whose contraction or relaxation can increase the pressure inside the lungs? Any other muscle? Well, I got many people are answering in this TV, and now I got an answer from this TV also. Raghuram, thank you, Raghuram, God bless you. And you thought it right, absolutely correct answer. Internal intercostal muscle is another muscle which can contract, IICM, which can contract and can increase the pressure. Okay. Well, any other muscle which contraction can increase the pressure? Any other muscle whose contraction can increase the pressure? Yes, guys. There is one more. Please say one more. Very, very good. These are the abdominal muscles. Very, very good. Let us call it abdominal muscles. Very good. Abdominal muscles, abdominal muscles. So, the muscles for inspiration are external intercostal and diaphragm, and these are the muscles for expiration. Why I'm not saying these? Because they are not working. They are just relaxing. So, a muscle should be actively participating in calling some muscle as an important muscle for that event. Remember that all the normal expiration occurs by relaxation of muscle. The normal expiration occurs by relaxation of muscle. My dear children, one is this point, and then pulmonary volumes. Yes, very, very important. If in this question, if you don't do this, you are going to make a mistake. Chances are there that you are going to make a mistake if you don't make these waves. Simple tides, one higher one and one lower one, more, deeper, taller, deeper. That's it. If you have to do this, otherwise you will not be able to think fast, number one, and number two, chances 10%, 5% chances of errors can be there, my dear children. This is a volume, because upward stroke means air going inside, and downward stroke means air is going out. And this height indicates a volume. So this height indicates the volume of air that we are inhaling, exhaling in normal breathing. Okay. Now this extra height that you see here, this extra height, this is the extra volume of air a person can inhale when he tries to inhale deeply. Okay, and similarly, this extra depth that you see, this volume of air a person can exhale when he tries to exhale deeply. So these are the three data that we have to remember. Okay, so this is a normal air going inside, outside. This is known as tidal volume. Tidal volume. Okay, what we are doing? We are revising pulmonary volumes. How many volumes are there? There are 4 pulmonary volumes. 4. Okay, tidal volume. This is known as inspiratory reserve volume. I for inspiratory, because upward stroke is inspiration. Similarly, this is called expiratory reserve volume. So these three volumes, and of course, even after that, there is some height we can assume, and this is the volume of air that is left in the lungs, even when we have got to the deepest point. Even after the forced expiration, still there is some volume of air in the lungs which is left. And this volume of air is known as the residual volume. And this is just, we have to imagine, and it is being calculated indirectly, but in clinical practice, we cannot calculate it directly. We can calculate the other three, TB, IRV, ERV directly. How we calculate? We calculate by Duckworth-Lewis method. Duckworth-Lewis means once we have these three values, applying the age and other things, age, sex, okay, ethnicity, and we can assume that what will be the residual volume after getting these three volumes. But at your stage, you have to learn the data. Tidal volume is half a liter. Inspiratory reserve volume is 2.5 to 3 liter. Expiratory reserve volume is 1 liter, and residual volume is 1.2 liter. Can you not remember this? These four, very, very easy. Half liter, 2.5 to 3 liter, 1 liter, and 1.2 liter. Whenever you have problem in remembering any data, digits, remember that this is nothing in comparison to in the next 4.5 to 5 years. What you are exposed to is something very, very horrible, literally horrible to remember. Big, thick books, and there won't be any Anurag Verma or there won't be any Gosalites there to make you revise the things again and again, and giving you so many of papers and all, so many of practice. Nothing like that. Even the teachers who come there, they are not very trained teachers. Okay, so that is a very horrible life we are, we are planning for, and everyone can Everyone can clarify this. Every year, many doctors become, once they can enter the medical college and they can clarify to you, me, everyone out there, we can also do. Okay, so we have to be prepared for remembering. Have confidence that I will remember. Okay, now applying these four volumes, we can have capacities, and those are called pulmonary capacities. Pulmonary capacities are 5 in number, and those capacities can be simply, now this will be in by doing the addition and subtraction. I told you now that one higher up, one lower one, and then this one. Okay, how much is this? Half liter. How much is this? 2.5 to 3 liter. How much is this? 1 liter. How much is this? 1 liter. And how much is left in the lungs? 1.2 liters. Okay. So half liter, 2.5 to 3 liter, 1 liter, and 1.2 liter. Now, what is the total volume of air a person can inhale by deep inspiration? Okay. Or either we can say by deep inspiration, can I also say that what is the maximum volume of air a person can inhale? Simple, same thing. Either I say total volume by deep inspiration, forced inspiration, or the maximum volume. Maximum, of course, comes with maximum effort. So maximum volume of air. So this is the sum of tidal volume. How much was this? Half liter. How much was this? 2.5 to 3 liter. So if this is 2.5 liter and this is half liter, this becomes 3 liter. So roughly 3 to 3.5 liter is this one, and this is known as inspiratory capacity. Similarly, guys, we can calculate the expiratory capacity, the maximum volume by forced expiration. Again, we can add half liter and 1 liter, and this becomes 1.5 liter. And what we call it? It is called expiratory capacity. Now, there comes an important capacity called vital capacity. What is the maximum volume of air a person can exhale by maximum inspiration? Okay, or what is the maximum volume of air a person can exhale by forced inspiration. So once you have inspired forcefully, so this much of air is in the lungs, and now you can exhale it out. So normally you are exhaling this complete one, okay? At sitting silently, this one. When you have taken a deep breath, then this one. But when you take a deep breath and we are giving maximum effort of exhaling it out, then actually we have done the transaction of this much of volume of air. Maximum inspiration and then maximum expiration. My dear children, how much that becomes? Okay, just add all of them. So what to add? 2.5, 0.5 and 1. 2.5 is the inspiratory reserve volume, 0.5 is the tidal volume, and 1 is the expiratory reserve volume. And whatever the number comes, you can say that to 4.5. That means 0.5 additional, because inspiratory reserve volume is not 2.5. It can be somewhere between 2.5 to 3 liters. This capacity is known as the vital capacity. Vital capacity. So this is a maximum transaction that is happening in the lungs. Maximum forced transaction, forced inspiration, then expiration. But still, it is not giving any idea how much of the air is left in the lungs. If somehow you have an idea of how much of air is left in the lungs, you will have a complete capacity, and we call it total lung capacity. My dear children, total lung capacity is a sum of all these: inspiratory reserve volume, tidal volume, expiratory reserve volume, and this one is the residual volume, Or we can say, we can add vital capacity, which is from here to here, and what is left is here to here, which is residual volume. So total lung capacity can be vital capacity plus residual volume. Total lung capacity is vital capacity plus residual volume. Or I can say that total lung capacity minus residual volume is equal to vital capacity. So anyways, they can give you either in terms of the data volume or in terms of these addition, subtraction. Okay, so we have seen the mechanism of breathing and this, and then an important part, how the oxygen and hemoglobin, they behave when we are like normal, doing the normal activity, then how much 100 ml of oxygenated blood can combine with 100 ml of oxygenated blood can combine with about 20 ml of oxygen. 100 ml of oxygenated blood has got 20 ml of oxygen, and similarly 100 ml of deoxygenated blood, 100 ml of deoxygenated blood, it has got around 15 ml of oxygen. That means there is not much difference of oxygen. And how much difference occurs from here to here? How much difference occurs? Only 5 ml oxygen. This much of difference has taken place. That means when the blood is coming to the tissues, in normal condition, it is only giving 5 ml. From which quota? From the quota of 20 ml. How much is given? 5 ml. What percentage? 25%. 25%. So in one circuit, when the circulation is going on, 25% of the oxygen is given to the tissues. But this is in normal resting condition. This is in a resting condition, and the person should be normal. Resting condition, and the person is a normal person. If a person is in vigorous exercise, during vigorous exercise, can you please tell me the requirement of oxygen is high or low? Answer is high. Can you please tell me the temperature of the body is high or low? When we are doing exercising, sir, the temperature of body is high. Okay. Can you please tell me the carbon dioxide content in the blood is high or low? Obviously, sir, CO2 content is high because there is active metabolism, more CO2 produced. Okay. So when these things are changed, when there is more carbon dioxide, tell me, when there is more carbon dioxide, the pH of the blood, the acidity of the blood, okay, I'll ask in this way because pH and acidity, I cannot ask both together. I'll ask acidity. When there is more CO2, there will be more acidity or less acidity? Now answer is more acidity because, yes, high acidity, very good, because CO2 makes carbonic acid, acid. Which acid? Carbonic acid. It will increase acidity. Okay. So whenever the exercise is done, CO2 is more, acidity is also high, and also the temperature, because actively metabolizing, temperature is also high, and then the same hemoglobin, which was actually carrying away how much percentage? Still, from 20 ml, how much is left? 15 ml, which means that it is 75% of the oxygen is carried away by the deoxygenated blood. My dear children, this is oxygenated blood, and this is the deoxygenated blood. 75% of oxygen what it has brought is still in the blood. Now we can obtain more oxygen because the requirement of the body is high, and we find that automatically the donation, the delivery, the dissociation of oxygen increases. The dissociation of oxygen increases, and the dissociation can increase up to 3 times. That means from 5 ml to 6, 7, 8, 9, 10, 11, 12, 13, 14, up to 15 ml. Up to 15 ml dissociation, guys. Dissociation. That means from the quota of 20 ml, 15 ml can be given to the tissues. My dear children, please understand this all, and please, whenever you do the diagram of the oxyhemoglobin dissociation curve, make sure you also notice these marks, and that is the reason NCERT has given you checkered tiles. It was not the intent to give you directly, only the shape. No, okay, shape is important, no doubt about that, but also some data you can obtain. So if the PO2 is 100 mm of mercury, if the PO2 is 100 mm of mercury, so there will be somewhere 50% marks, and this is 40 mm of mercury, and there will be 30 mm of mercury. So 30 mm of mercury, 40 mm of mercury, and 100 mm of mercury. Similarly, you can put 100%, 50%, and these are, so 25% saturated, 50% saturated with oxygen. Who is saturated? Hemoglobin. 75% saturated and 100% saturated. This is oxygen. This is PO2, oxyhemoglobin dissociation curve. Already we know the shape of the curve is sigmoid. Shape of the curve is sigmoid. Okay, so normally 100 mm of mercury, there will be 100% of the saturation. But in 40 mm of mercury, where should be the point? It should be 40 mm of mercury. That means the venous blood, it is still having 75%. See, in the venous blood, it is still having how much? 75% in the venous blood. Okay, 75%. Now, in the 41, 30 mm of mercury, it is still 50%. Please remember, this is a fact. 50% of hemoglobin is filled with oxygen or is bound with oxygen. So this type of curve that can be seen, this type of curve or sigmoid curve, these data, so I'll put these data, I'll make this thin. This is 50% marks, and 50% marks come at 30 mm of mercury, 75% comes at 40 mm of mercury, and 100% comes at 100 mm of mercury, 100 mm of mercury here. Okay. Whenever the carbon dioxide acidity temperature, all these increase, you will find that the same curve will be now shifting towards the right. The same curve can start shifting towards the right, okay? And this all depends how much of carbon dioxide is high, how much of acidity is high. And from 75% that you notice here, this can even go to 25%. I mean to say, in the vein, I use blue color. In the vein, this is venous blood. In the vein, earlier, how much was there? 75% saturated, and now you can see this can come up to 25% saturated. The same hemoglobin in the vein, earlier 75% oxygen was remaining, and now 25% oxygen is remaining in the veins. That means earlier 25% dissociation has taken place, but as it is increasing carbon dioxide acidity and temperature, as the curve is going towards right, and we find that now it is 25%. If I apply this knowledge here, this is a normal resting condition, 20 ml, 5 ml. But when a person is doing vigorous exercise, if a person is doing vigorous exercise, then this 5 ml can become 15 ml. And this becomes 25%, even though then also it is not, and this becomes 5 ml. That means the story is reversed. From 25%, it becomes 75% dissociation. Hemoglobin can change its structure. Hemoglobin can change its conformation, can change its nature by increasing carbon dioxide acidity and temperature. This is known as the Bohr's effect. This effect changes in the hemoglobin nature, hemoglobin affinity for oxygen, changes in the hemoglobin affinity for oxygen is called the Bohr's effect. However, if this effect is not named in the NCERT, they may not give this particular name if it is not there, because we know it's not there. But the concept should be known, okay? Here one concept has arisen, and that concept is called the concept of P50. P50 is that PO2 at which hemoglobin is 50% saturated. And my dear children, how much is the P50 in normal resting condition? P50 is 30 mm of mercury. P50 is 30 mm of mercury. Can you please tell me what will happen to P50 as we increase carbon dioxide acidity and temperature? The P50 options are P50 increase, P50 decrease, option number 3, P50 remains same, because it is an integral characteristic of hemoglobin and it will not change. Option number 1, question is, when CO2 acidity temperature increase, what will happen to the P50 value? Option number 1, P50 will increase. Option number 2, P50 decrease. Option number 3, P50 will not change. The correct answer is P50 will increase. Bacha, just see here, P50 is also increasing. As the curve is going right and right, P50 is increasing, P50 increase. That means at more PO2 required to fill the box 50%. That means the affinity is not that great. And this can be remembered, CAT can be remembered as Pilly cat. CAT cat. Okay, guys. About the regulation of breathing, remember that we have a rhythm center in medulla oblongata. 100% type of questions. Where is the rhythm center? It is present in medulla oblongata. Which center is present in pons varolai? Pneumotaxic center. Which part of the brain control the breathing primarily, or control the normal breathing? The two parts, medulla oblongata and pons feruli. Okay, what is the pontine center? Pontine means pons wala. What is the pontine center or pneumotaxic center due to the breathing rate? Will it change? Will it increase, decrease? If the firing of the pneumotaxic center is high, the breathing rate becomes higher. If the firing is at a lower frequency, and the breathing frequency will be lower. If pneumotaxic center firing frequency is high, the breathing frequency per unit time, the breathing will be higher. That means more number of breath per minute, more number of breath per minute. Hello. 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 हाँ जी बोलिए सर बोलिए. थैंक यू सर, थैंक यू सो वेरी मच. मैं भी उसमें था, मैराथन क्लास. मैराथन. ओके सर, ओके. थैंक यू. So the pneumotaxic center, it controls the rate and depth of breathing. Rate and depth of breathing. This is an important line one should remember. Pneumotaxic center control the rate and depth of breathing. Talking about the body fluids and circulation. Starting with the body fluids, the blood and lymph are the body fluids. The pH of blood and of course the lymph. Mainly the blood pH should be known. It is 7.4 is the pH of blood. If the blood pH changes, there will be a lot of problems that can happen. Just now we have seen if the pH decreases, we see that the oxygen carrying capacity of the blood also changes. If pH decrease, oxygen carrying capacity also decrease. We have seen CAT, acidity, the curve shift towards right, same thing. If the pH decrease, oxygen carrying capacity also decrease. That is one thing. In body fluids, the different components of the body fluids, the blood and lymph, components of the blood, cells and matrix. Matrix called plasma. Plasma has got what? Plasma has got protein. 6 to 8% of plasma are plasma proteins. In plasma proteins, what are the main plasma proteins? The main plasma proteins are albumin and globulin. Albumin smallest and then globulin. But then a small percentage of largest plasma protein called fibrinogen is present. Fibrinogen. It is a soluble protein, fibrinogen, and it can be activated to fibrin in the clotting pathway. So fibrinogen is a soluble protein, largest protein, and this helps in blood clotting. If you exclude fibrinogen from the plasma, the remaining fluid is called serum. Normally, the word serum is used with respect to the antibodies, okay? Now this TV has started working. That's really good. Now I have both the TVs working. It's really good. My dear children, so please understand this. Now, the clotting pathway, what are the things that you already know in the clotting pathway? In the clotting pathway, we should know the 4 clotting factors, which is clotting factor number 1, second, third, and fourth. First, second, third, fourth. Okay, and this is fit pants, tight capri. Fit pants, tight capri. Fit pants, tight capri. Okay, so we have the first one is fibrinogen. Just now I have taken this name, a plasma protein, largest plasma protein. Now pants is prothrombin, prothrombin. Fit pants, tight, thromboplastin, thromboplastin or tissue thromboplastin, thromboplastin, also called tissue thromboplastin. It is a tissue factor. Remember, it is a tissue factor, thromboplastin. And then we have the fourth one, calcium ions. Okay, I again say fit pants, tight capri. Fit pants, tight capri. Remember these four factors. Then remember that there are two pathways. One is an intrinsic pathway and one is an extrinsic pathway. And by different mechanisms, these pathways are stimulated. Intrinsic pathway within the blood vessel, extrinsic pathway where there is some injury and the blood vessels have opened up, okay, and the blood is coming out, and then extrinsic pathway will try to fix it. Immediate fix, remember that. Immediate fix of any leakage, which blood cell help in this immediate fix? Immediate fix is done by number one, erythrocytes, number two, leukocytes, number three, fibrin, number four, platelets, which is responsible for immediate fix. Fix matlab make it all right. Immediate fix, platelets. Very good. Answer is blood platelets. These platelets, they just make a plug there called platelet plug. And for example, it's a blood vessel and the leakage has started. The platelets will start accumulating here and will plug this, and the bleeding is stopped. And then in this time, what is happening? Those extrinsic pathway is now activated. And this pathway will ultimately lead to the formation of an important protein complex, enzyme complex. This enzyme complex is not a single enzyme. It is not a single enzyme. This enzyme complex is a complex of enzymes, and this is a key enzyme, and that is called thrombokinase. Thrombokinase. By both the pathways, the intrinsic pathway as well as the extrinsic pathway. Intrinsic pathway and extrinsic pathway ultimately lead to formation of an enzyme complex. Guys, this is not a single enzyme. This is a complex of enzymes. It's an enzyme complex. This can transform, this can convert prothrombin to thrombin, prothrombin to thrombin. Pro means inactive, thrombin means active. Thrombin can convert, yes, any idea? Fibrinogen to fibrin, soluble to insoluble, monomer to polymer. Guys, fibrinogen is soluble monomer, fibrinogen, soluble monomer, fibrin, it's insoluble polymer. Please remember, and once these fibrin threads are formed, those threads, they form a meshwork, and in that meshwork, of course, in the beginning it is very wet, it dries up, and that meshwork trap the blood cells. You know that maximum blood cells are RBCs, so majority of the RBCs are trapped. And those RBCs also give a reddish color to this fibrin, clot. This coagulum or clot, this is called coagulum or clot, and this is called blood coagulation or blood clotting. At every step, what is the main thing required at every step is, yes guys, at every step we require calcium ions. And in some of these pathways also, we require calcium ions. That means if somehow I capture the calcium ions, I can stop the whole pathway. Therefore, many type of anticoagulants are available which can capture the calcium ions, Hope you understand this. Many type of clotting, anticoagulants. For example, anyone, just an idea, I know it is not given so directly anywhere in the NCERT. If at all you know any anticoagulant which capture calcium ions, come on. Here guys, come on, answer. 10 minutes break, pakka. Because here tea has come, I also want to. Heparin, this one. It's not heparin, it's, you have heard about EDTA, ethylene diamine tetraacetic acid. Ethylene diamine tetraacetic acid. Okay? Please remember this. And yes, heparin is an anticoagulant, but heparin mechanism of action is not capturing calcium ions. My dear children, hope you understand this. This is blood clotting. Maximum questions on thrombokinase. Important, very, and 5 times more, very important thing. Very, very important. Teacher will feel good in making questions. And in coming, our test, our grand test, I will be giving you 1 or 2 questions on any of these, okay? So now I'm giving you a 10 minutes break, and this time it is 10:30, and at sharp 10:40, I will start, or 10:41, I will start speaking again. 10:41, meeting at 10:41. We are meeting at 10:41. The cardiac control center is present in medulla oblongata. This just appeared to my mind. You should remember that. Then about the different phases of cardiac cycle, let us make a very simple illustration of different phases of cardiac cycle. The smallest phase is atrial systole, 0.1 second, then ventricular systole, 0.3 second, and the joint diastole, 0.4 second. The atrial diastole begins from this point. After the systole, the next will be atrial diastole. So till here is the atrial diastole. The ventricular diastole begins after the ventricular systole and will continue till the next beginning of next ventricular systole. You can see that both green and white are here. That means for both the chambers, therefore joint diastole. But individual, if they ask you, the atrial systole, the duration of atrial systole is 0.7 second. Very simple, you can say like that. 0.8 second is the total duration and 0.1 second is the time required for atrial systole. So the rest of the cycle is atrial diastole. Similarly, 0.3 is the ventricular systole. 0.8 second is the total duration. 0.8 minus 0.3 becomes 0.5, like that. Otherwise also, if you just try to calculate from here to here, you will come 0.3 plus 0.4, give you 0.7. And From year to year, this is 0.4 and this also 0.1, and therefore this becomes 0.5, okay, 0.5. That means ventricular diastole takes 0.5 seconds. For half of the duration of cardiac cycle, the heart remains as a common chamber. For half of the duration of cardiac cycle, there is joint diastole. The heart remains as a common chamber. Now here, the very straightaway questions that can be asked. Lub, the sound 1, comes at the beginning of ventricular systole, and the dup, the sound 2, appears at the end of ventricular systole. The duration between lub and dup is 0.3 seconds, and the duration between dup, the sound 2, and lub, the sound 1, is 0.5 seconds. Because from between this is 0.3, now once you hear this sound, and the next lub is this duration, and therefore it is different, 0.5 seconds. And also ventricular filling. Please understand that ventricular filling takes place in 2 phases. Ventricular filling takes place in 2 phases. One is a passive phase, passive filling phase, passive, and it is a rapid filling phase. Passive and it's a rapid filling phase. And one is an active filling phase, and this is a slower filling phase, okay? So the rapid filling phase, this occurs during joint diastole. 70% of the ventricular filling takes place in joint diastole, then 30%, and this takes place active one during atrial systole, and 30% is attributed to atrial systole. Please keep this point in mind. So passive filling phase, that means maximum volume of the ventricles is filled during diastole. That means without the systole of the atria. Remaining 30% by atrial systole, the contraction of atria. Remember that in diastole, any chamber receives the blood. In systole, any chamber exit or pump out the blood. The blood pumped out from each ventricle in one cardiac cycle. When one cardiac cycle over, from each ventricle, the blood comes out is about 70 ml. 70 ml from each ventricle. Now, this is per cardiac cycle, per heartbeat. If number of heartbeat in 1 minute is X, so we have to multiply this 70 ml into X. Normally, the heart rate or number of heartbeat rate is 72 per minute. And the volume of blood pumped by each ventricle in one heartbeat is 70 ml. So the volume of blood pumped out of each ventricle in 1 minute will be heart rate, that is 72 into 70 ml, which is roughly 5 liter. That means from each ventricle, 5 liter of blood is pumped out in 1 minute, 5 liter per minute. My dear children, please note here that there are some names to this. For example, the 70 ml is the volume of blood pumped out in one heartbeat. In one heartbeat, this is known as stroke volume. It is known as stroke volume. Stroke volume, and this in 1 minute, this is known as cardiac output. Cardiac output. Cardiac output. Please understand the stroke volume and cardiac output. And the stroke volume is 70 ml. That means 70 ml of the blood is pumped out of the heart in 1 minute. In 1 minute, in one heartbeat, 70 ml of blood is pumped out of heart. Suppose if this is the heart, that means the amount of blood coming out of heart per heartbeat is 70 ml. Then can we say how much blood enters into heart per heartbeat? Same 70 ml, because the heart gives what it gets. If it is getting 70 ml, it will give out 70 ml. If you increase this will also increase. In the ventricles, only 50 ml of the blood is remaining. 50 ml. 50 ml of the blood is always remaining in the ventricle. Atria, there is no trapment of the blood, but in each ventricle, 50 ml of blood is filled. That means if 70 ml comes, the end diastolic volume will be 70 plus 50. How much it will be? It is 120 ml. End diastolic volume. And after the systole, the end systolic volume is remaining kitna? 50 ml. That means the difference of this is called the stroke volume, and this is 70 ml. So heart gives what it gets. And of this 70 ml, which will receive more blood? Right atrium, left atrium, right ventricle, left ventricle. Which of the following chambers receive maximum blood? Okay, and one of the answer is all chambers receive equal blood. Correct answer is all chambers receive equal amount of blood. Whether it is atria or ventricle, irrespective of that, they receive equal blood. For example, we know that 70 ml is pumped out in one heartbeat. Okay, that means that 70 ml enters into the right atrium. Okay, 70 ml enter into the right atrium. The same 70 ml come into the right ventricle. The same 70 ml is going to the lungs, and the same 70 ml is coming to the left atrium. Same 70 ml. And the same 70 ml come to the left ventricle, and the same 70 ml pumps out. That means all the four chambers, they are receiving only 70 ml. All the four chambers, equal amount of blood. All four chambers receive equal amount of blood. My dear children, please understand that the normal heart rate, which should be without any nerve supply, is about 110 to 120 heart rates per minute. I repeat, normally our heart can, I'm not saying it is beating, but can beat 120 up to 120 heart rates per minute, beats per minute. But it is not that much in normal condition. Actually, there are always brakes present, brakes. And these brakes are applied by parasympathetic nerves. Sympathetic nerves. If someone asks you which parasympathetic nerve or which nerve is carrying parasympathetic fibers, your answer is, sir, it is vagus. Vagus is carrying parasympathetic fibers to the heart. And this is applying a brake on the heart rate. If these brakes are not there, then the heart rate will be more than 100. But in the presence of the brake, the heart rate is about 70 to 75, somewhere in between per minute, these many heart rates. If parasympathetic nerve supply is cut, then what will happen? The heart rate, what will happen? Heart rate will, because brakes are withdrawn, if brakes are withdrawn, heart rate will increase. The same answer is given if vagus nerve supply, if vagus to heart is cut. Vagus to heart is cut, then also the same thing, because vagus is carrying parasympathetic fibers. My dear children, sympathetic nerves are increasing the heart rate and blood pressure. So sympathetic nerves, they are increasing the heart rate and increasing the blood pressure. Sympathetic nerves, okay? Please understand here that there is no such special name given to any cranial nerve which carry the sympathetic fibers. Sympathetic fibers are directly coming out from thorax and lumbar, thoracolumbar. Sympathetic is thoracolumbar outflow. So directly, there is no such name to be remembered here, no cranial nerves. Also remember that the same effect can come if we use any of the neurotransmitter as a hormone. If any hormone is used, for example, epinephrine. Epinephrine can also increase the heart rate and blood pressure. And the same effect can come by norepinephrine also. Norepinephrine. It will also bring about the same sympathetic effects on the heart. So epinephrine and norepinephrine, they increase the heart rate and blood pressure. And the vagus nerve decrease the heart rate and blood pressure. And if vagus is cut, the heart rate will shoot up. Because in the presence of vagus, even without the stimulation of vagus, there are some brakes applied to the heart rate. Let me tell you, normally the heart is designed by God to beat more than 100 times per minute. But in the presence of the vagus stimulation, vagus is like a brake and the heart rate is not more than 70, 75, 80. Please keep these points in mind. Hope you understand all these. Hope you understand the sympathetic, parasympathetic. These are the important places. A very, very important question can be asked. If parasympathetic nerve supply is cut, a very, very important question, I'll give you very, very important question. If the parasympathetic nerve supply is cut, the answer is heart rate will increase. And similarly, any of these, either stimulation of the sympathetic nerves or we inject epinephrine, norepinephrine, it will increase the heart rate and blood pressure. Let me tell you, when the person dies in hospital, we perform CPR. CPR is a set of procedures which are done as a ritual to make the body alive. And sometimes, if the luck is there, the body becomes alive again. So you might have seen people are doing the cardiac massage, just pressing the chest of a dead person, immediately dead, or applying very high voltage current on both the side of the chest so that the body can even jump up from the bed. Okay, it is also part of the CPR. You might have seen that people are just giving breath either mouth to mouth or by an instrument called embu bag. At the same time, the nurse take an injection and just press it, on the chest. One or sometimes two injections. What is that she is injecting? She is injecting epinephrine. She is injecting epinephrine on the chest because epinephrine can increase the heart rate and blood pressure. Okay. My dear children, this is one thing that you should remember. Another point is the sequence of the nodal tissue. The nodal tissue is autoexcitable, which comprise of SA node, then we have AV node, then we have AV bundle, and bundle branches and its branches, AV bundle and its branches, okay. And after that we have Purkinje. Fibers. And this is the order of the passage of the signals. What is signal? Actually, signal is nothing but action potential. Remember, here you will find action potential. Action potentials. Very good. Action potentials. And all of them can generate action potential. Remember, all of them can generate, but the frequency is different. For example, at 40 times per minute, AV node is generating action potential and SA node is generating about 70 to 75 times per minute. And that is maximum for any of these four places. Maximum is 70 to 75 times. Okay, and therefore the one which is maximum takes away all the other, and it is the sinoatrial node which determines the heart rate, and therefore it is called the pacemaker of the heart. Sinoatrial node, pacemaker of the heart. Okay, guys. But then remember, all of them can generate the action potential. Okay. Lastly, one point here to understand is the electrocardiograph. The electrocardiogram comprises of these few wave patterns. Okay. So there is a first positive wave called P wave, then the negative wave, and then again a positive spike, and again another wave called the T wave. Generally, this is what we obtain in a general electrocardiograph. Electrocardiography is the procedure wherein the body is plugged with different electrodes, the wires. So we can put a wire on the right wrist, a wire on the left wrist, and a wire on the left ankle. Okay, right side, left side, right side, left side, on right and left wrist and left ankle. Okay, we can apply these three wires and we can start the ECG. But nowadays, this is a standard of classical ECG. These days, about 12 wires are used. Yes, you heard right, 12 wires. These days, up to 12 wires can be used. Actually, we call it 12, but then one of them, one is applied here, total 4, and one keeps on moving. We keep on moving it 6 times like that. And from one place we obtain 2, one place we obtain 1. So we call them 12 leads. With every side is called a lead. I mean to say, please remember the classical wire setting. This can be asked in the examination. And also remember the name of these waves, PQR, S, and T wave. And why these waves are created. The P wave is because of the atrial depolarization. Atrial depolarization. The complete QRS complex is for ventricular depolarization, my dear children, and the T wave is for ventricular repolarization. Ventricular repolarization. I mean to say, for atrial repolarization, we don't have any separate wave pattern. Listen once again. For atrial repolarization, we don't have any separate wave pattern. Yes, for ventricular repolarization is the T wave. But the problem begins when is the systole. Remember, what did I say? When is the atrial systole? My dear children, atrial systole begins little after the center of the P wave and completes by the time R wave is also over. So from this point to this point, this complete is this duration. This is atrial systole. Earlier, what did I say? Atrial depolarization. But systole takes place little later than depolarization. The red color is atrial systole. Immediately after that, we have ventricular systole. And ventricular systole begins just after this. Do you know when the ventricular systole gets over? It gets over when the T wave is also over. So this is ventricular systole. Ventricular systole. And tarvata, we will have ventricular diastole. and also atrial diastole. So joint diastole, atrial systole, and this is the ventricular systole. Tarvata, we have joint diastole. Keep these points in mind. Also, if you remember, we have made the cardiac cycle wala circle. The cardiac cycle circle, this one. There is a first, heart sound at the beginning of ventricular systole, and the second heart sound at the end of ventricular systole. Now, when we apply this to ECG, then where will be the sound 1? Sound 1 begins at this point. This is the point where we have sound 1, the lub sound, okay? And this is the point where we have sound 2, S2, or the dub sound, sound 2. Ventricular systole beginning and end, S1 or lub and S2 or dub. End of T wave marks the end of ventricular systole, but formation of T wave marks ventricular repolarization, but the end of T wave marks the ventricular systole, or simply heart systole. Or ventricular systole begins just after the Q wave. I repeat, just after the Q wave, ventricular systole begins. All these points that just after the Q wave and at the end of T wave, these points are mentioned in your textbook. Therefore, you should remember that, make it simple line diagram, put sound 1, sound 2. Never forget that how this sound 1, sound 2 are put. If we make a circle and we have done, for example, atrial systole, ventricular systole, and joint diastole. Therefore, I said that after that we have joint diastole. And therefore, I said that here and here, and here, we have sound 1 and sound 2. Sound 1 is lub and sound 2 is dub. My dear children, we have another chapter called the excretory system, or nitrogenous wastes and their elimination. Okay, so we have the nitrogenous wastes and their elimination, So we have 3 types of nitrogenous waste. We have ammonia, urea, and uric acid. Ammonia is at this end and uric acid at the other opposite end. Uric acid requires minimum amount of water for its elimination, uric acid. Whereas ammonia requires maximum. Uric acid requires minimum. Please keep these points in mind. Just one. ये तो साथ चलता है ना. वॉइस वगैरह सही आ रही है क्लास में? वॉइस नॉर्मल?
अच्छा।
Guys, just a second. I am coming back. Just a second. ग्यारह 40 पर 11:40 पर एक ब्रेक दे देंगे 5 मिनट्स का, है ना? वो लास्ट ब्रेक होगा 5, बच्चे बोले 10 मिनट्स ताकि फ्रेश हो जाएंगे बच्चे। 10 मिनट्स दे देंगे। एक ब्रेक हमने दे दिया, हमने इसमें टाइप कर दिया था, बोर्ड पे लिख देंगे, ठीक है, नो प्रॉब्लम। My dear children, ammonia requires maximum water and uric acid requires minimum water and urea requires moderate amount of water. The evolution of urea and uric acid are for terrestrial adaptation. Ammonia is mainly found in animals in aquatic habitat. So mainly it is the aquatic animals which are ammonotelic. Okay, so this list is just for your revision. Yes, this is an important list. Okay, keep it in mind. If you have NCERT open with you, you can just put some, you can just fold the page a little bit, the corner of the page, so that again you have to come back and revise the list. Again, you have to come back and revise the list. Then the urine formation. The urine formation takes place by 3 methods, which is ultrafiltration, selective reabsorption, and tubular secretion. By 3 methods, urine formation takes place. Dear children, remember that ultrafiltration is the least selective process. In fact, it is almost, we can say, it is not selective process at all. It is not a selective process or least selective among the 3. Okay. Ultrafiltration leads to the GFR of 125 ml per minute. That means in 1 minute, 125 ml of the filtrate is formed by ultrafiltration. Now, this is a big amount. Of course, it is by both the kidneys, 125 ml. This comes out to be about, okay, 1,80,000 ml per day, and this is a big amount. My dear children, the urine produced per day is 1 to 1.5 liter. This is the urine produced per day. That means where this goes back to the blood in the process of reabsorption. But then even though something which is not filtered out is secreted again by tubular secretion. It is also helping in maintaining the pH and ionic balance of the blood, the pH balance and the ionic balance of the blood. So ultrafiltration, selective reabsorption, tubular secretion, these 3 steps leads to formation of urine. Ultrafiltration filters everything except large proteins and blood cells. So what is this filtrate? It consists of this ultrafiltrate or glomerular filtrate. It consists of the components of the blood except the cells and proteins. Everything of the blood can be filtered. That means it is a least selective process. That means it has got good things as well as bad things. The good things have to be reabsorbed back. And now let's see where are the reabsorptions occurring. The maximum site of reabsorption is the convoluted region of the near the glomerulus. This convoluted region is known as proximal convoluted tubule. Why proximal? Because this is the most near the glomerulus. Okay, if I show it like this, so you have this is Bowman's capsule, and starting from here we have a convoluted tubule, highly convoluted proximal convoluted tubule. This is near the glomerulus, therefore proximal. Proximal means near, proximal. And you have these loop of Henle, hairpin loop or loop of Henle, hairpin loop, hairpin, I think hairpin, your age might not have seen these long hairpins. Hairpin loop. Again, we have convolutions, means these coiling, folding, and then it becomes part of the common duct or collecting duct. So this is actually a collecting duct. Okay, so this is collecting. In this part, we can say what the fluid inside is called urine, and before that we can call it a filtrate. These parts, and here we have the glomerulus. This is a glomerulus. This is PCT. Maximum reabsorption occurs at PCT. What all gets reabsorbed? But everything gets reabsorbed means 100% of nutrients. 100% of nutrients. Nutrients means glucose and amino acid. 100%. Okay. About 70% of water and electrolytes. About 70% of water and electrolytes are absorbed, or in fact reabsorbed in this part. Reabsorbed. And when we say electrolytes, it comprises of sodium, potassium bicarbonate. Sodium, potassium bicarbonate. These are the main electrolytes. Now the descending limb of loop of Henle, this is only permeable to water. The descending limb is only permeable to water. Let's understand, let's remember this. The descending limb is only permeable to water, and the ascending limb is only permeable to salt. The ascending limb is only permeable to the salt. And the main salt here, which I say it is permeable to, it is NaCl, okay? Now this differential permeability as well as this long hairpin type of system and the counter current existing between them, the counter current means in this segment, the filtrate is going in this direction. In this segment, the filtrate is going in opposite direction. So opposite flow is called counter current flow. And this is also important for maintaining high osmolarity in the medullary interstitial fluid. Deep down the medulla, the special type of nephron, which are called the juxtamedullary nephron, they help. But if a question comes, what is the main role of loop of Henle to maintain osmolarity in the medullary interstitial fluid? Now, in this part, the sodium reabsorption takes place. At the same time, the potassium secretion takes place. Sodium reabsorption, potassium secretion. And this is a place where water reabsorption can occur. And this is in presence of a hormone called ADH, antidiuretic hormone. Can anyone tell me what is this ADH is? ADH is a protein hormone or it is a steroid? Antidiuretic hormone is a peptide hormone or a steroid. One in this TV, guys, It's a protein hormone. Very good. It's a protein hormone, antidiuretic hormone. In which gland it is synthesized? Antidiuretic hormone, which gland it is synthesized? Heart. Hypothalamus, it is synthesized in heart में बेटा वो ANF, that is atrial natriuretic factor, atria, heart. This is in hypothalamus. It is a hormone released from posterior pituitary, no doubt about that, but it is synthesized in hypothalamus. Antidiuretic hormone. The last question about that is, what is another name of ADH, antidiuretic hormone? What is another name of ADH? Vasopressin, perfect. What is the role of ADH on blood pressure? The role of ADH on blood pressure. Role of ADH in blood pressure, increase, decrease, no change. ADH on blood pressure, increase, decrease, no change. It will increase, as the name indicates, vasopressin, that means constriction, vasoconstriction and pressure, blood pressure will increase ADH. ADH both by reabsorption of water and also directly by vasoconstriction. By both the ways, it helps in increasing the blood pressure. Okay, and in any segment, if minimum reabsorption occurs, answer should be ascending segment or ascending limb of loop of Henle. In the ascending limb of loop of Henle, minimum reabsorption takes place. I repeat, minimum reabsorption takes place in the ascending limb of loop of Henle. Another point, conditional reabsorption occurs in. Conditional reabsorption occurs in. Conditional means if on the condition, if there is a lack of blood volume or there is less blood pressure, any of these conditions, either the blood volume is less, blood pressure is less, any of these conditions, conditional reabsorption takes place in the distal convoluted tubule, DCT. Okay, guys. So just keep this point in mind. This is all main point about the urine formation, the urine concentration, okay, and the main concentrating segment in the urine, the main concentrating segment is this part. But this part will only concentrate the urine provided that there is high osmolarity in this part with the help of loop of Henle. So I'll give you a couple of questions and answer myself. I'll tell you, urine concentration, why we require to concentrate the urine? We require to concentrate the urine to minimize the loss of water. Is it required every time? No, it is required only when we have scarcity of water, when we have somewhat lesser water. What are the conditions? How we lose water? Water from the body can be lost mainly by urine, then by sweating, because sweat also contains water. So mainly by urine, water can be lost from the body and by sweating. In what ways we gain water? Maximum water is gained by food or drinking, and some water by metabolic water. Metabolic water means which is formed during metabolism. So if the loss of water is more than the gain of water, at that time, there is a necessity of urine concentration. In which part the real concentration of urine takes place? Concentration of the urine occurs where actually the urine is. For example, in the collecting duct. In the collecting duct, reabsorption of water can take place and it can concentrate the urine. What are the preconditions required for water reabsorption from the collecting duct? What are the preconditions required for water reabsorption from the collecting duct? My dear children, the conditions required for water reabsorption from the collecting duct are these two. Conditions required. One is the supply of ADH hormone, which makes this segment permeable to water. Yes, you heard right. Collecting duct is normally impermeable to water. In the presence of ADH, it becomes permeable to water. ADH. Okay. Another condition required for this reabsorption is a high osmolarity in medullary interstitium, or I can call it osmolarity gradient. Osmolarity gradient. Different grades of osmolarity. In the medulla, in the renal medulla, different grades starting from 300 milliosmol per liter up to 1200 milliosmol per liter. Up to 1200 milliosmol per liter. 300 to 1200 milliosmol per liter. Different grades of osmolarity, and this is called the medullary gradient or osmolarity gradient. This is also required for the reabsorption of water. And the last question is, or the next question is, what maintains osmolarity gradient in the medulla? And here comes a mechanism called counter current maintains osmolarity gradient in the medulla. Counter current. But counter current in what? Answer is counter current of filtrate in the loop of Henle and counter current of blood in the vasa recta. Do both have almost the same structure? Yes, I mean to say both are running anti-parallel. Okay, so both are forming the hairpin loop. Long, they are very long, going to the deepest part of the medulla and taking a hairpin loop. That is called counter current mechanism. So where it operates in loop of Henle as well as in the as well as in the vasa recta. My dear children, please remember that they both have different functions. For example, the loop of Henle can maintain high osmolarity, can reabsorb salt in the medulla. So in the medulla, with the help of loop of Henle, because the ascending limb of loop of Henle is permeable to salt, and moreover, there can be active reabsorption. Active reabsorption can take place in the thick segment. That means lot of salt can be concentrated here. Lot of salt can be concentrated, and 50% by the salt and 50% by urea. That means if I only show you the medulla, the renal medulla, in the medulla there is a gradient, you know, from 300 to 1200, and there is a mixture of urea and NaCl, and each component 50-50, each component 50-50, 50-50. Okay, so whatever that you see, for example, 1200, out of the 1200, 600 is by urea and 600 by NaCl. Loop of Henle is sufficient to maintain NaCl in the medulla. The vasa recta help in preserving this because no doubt it is permeable. It will take NaCl into it. The descending limb of vasa recta will take NaCl. The descending limb of vasa recta will take NaCl into it by simple diffusion. But the ascending limb of vasa recta, again diffusion takes place and NaCl out. NaCl in, NaCl out. So NaCl remains in the medulla. Vasa recta is doing other works. You know that because you might be thinking, sir, it is coming down, it is taking some salt and giving water. Why did it ascend up? Why? It will give the salt and take in water. So water in and salt, that means exactly opposite to it is happening in the counter loop, in the counter limb. Answer is, well, then what is the need of vasa recta? The need of vasa recta is Every part of the tissue, every tissue in the body, every organ in the body requires blood supply. Blood is amritam. Blood brings gases like oxygen. Blood brings the food and takes away the waste. Blood is very important. And that's the reason other works can be done by this vasa recta. But it is not carrying away, it is not washing away NaCl. Medullary NaCl remain in the medulla. That means vasa recta help in preserving medullary gradient, preserving. And medullary gradient plus dash requires for urine concentration or urine concentration in collecting duct. Medullary gradient plus dash in this TV. If anyone can write the answer in this TV, please. Come on. Medullary gradient plus what? Answer, beta. I just said one thing, just. Yes, I take that, no doubt. You can say vasopressin, not a problem. Vasopressin, perfect. ADH or vasopressin, very, very good. So no need to write ADH again, same thing. Even if you write vasopressin, I will take that. Very good. My dear children, urine concentration, direct lines can be asked from the NCERT about the exchanger and also multiplier. We know that loop of Henle is countercurrent multiplier and vasa recta is countercurrent exchanger. Please take these points in mind. And regarding the kidney, yes, I'm telling you, there is a short question in RAAS system. And you know what is this RAS? RAAS. RAS is renin, angiotensin, aldosterone system. Renin, angiotensin, aldosterone system. After a break of 5 minutes, it's not a break actually. It's a emergency break. Okay. After a break of 5 minutes, I'll be resuming. If you want, you can take a break. You can also go for 5 minutes. So, in 5 minutes, coming in 5 minutes, coming in 5 minutes. Okay. Let me start with the renin-angiotensin-aldosterone system. Very first thing that you should know about this system is what actually the target of this. The target of this system is to increase the blood pressure, because this system is activated when the blood pressure is decreased. But why the kidney is maintaining the blood pressure? Why it is the work of the cardiovascular system? Why the kidney is concerned with that? But remember, if the blood pressure is changed, the GFR also changed, the glomerular filtration rate also changes if the blood pressure changes. Actually, kidney is maintaining its GFR. Clear? Okay. So all renin, angiotensin, aldosterone, either of these three or all three of them, they are increasing the blood pressure. For that, I hope you remember juxtaglomerular cells. These are the specialized smooth muscle cells of afferent arteriole. I repeat, specialized smooth muscle cell of afferent arterioles. Afferent arteriole, glomerulus, and efferent arteriole, and the smooth muscle cells they have become larger also, and they have become blood pressure measuring machines. So these are actually the blood pressure sensors. They have become blood pressure measuring machines. We have also natural BP measuring machines. What is the name given to the blood pressure measuring machine? The name given is sphygmomanometer. Sphygmomanometer. But we have these natural one. They will not give us any data or any manometric reading. If the blood pressure is decreased, they will become activated and they will release a substance called renin in the blood. Renin. Remember that this is a different renin than the renin, R-E-N-N-I-N, which was there in the infant's stomach, infant's gastric juice. Renin is released. Moreover, the distal convoluted tubule, let's make DCT, distal convoluted tubule is somewhat going perpendicular like this, DCT. And when the BP decreased, obviously the GFR is also decreased. The GFR will be decreased, and obviously this will make some changes in the water as well as salt reabsorption. There will be less salt in the filtrate, and less salt in the filtrate activate next part of this system, and this is known as macula densa. You know, macula means spot, densa means dark. Macula densa, I repeat. How this macula densa gets stimulated? Remember, when BP is low, right? GFR low, and therefore there are some changes that occur in the reabsorption of sodium. Actually, whenever the PCT finds that the GFR is low, it will reabsorb salt more. It will reabsorb salt more. So the salt concentration, the NaCl concentration in the PCT will be low, because PCT, proximal, has reabsorbed it more because it finds low GFR. As a result of that, Na concentration in the DCT is low, in the filtrate is low, and therefore it stimulates these parts. This is known as macula densa. Macula densa in turn stimulate the same JG cells. So these are the JG cells, juxtaglomerular cells. Macula densa also activate them. Directly they are activated by low BP. As a result of that, the renin is released into the blood. Renin is an enzyme. It can catalyze the conversion of angiotensinogen into angiotensin 1. Angiotensin 1 is further converted to angiotensin 2. By another enzyme known as, very simple, ACE, angiotensin converting enzyme. Angiotensin converting enzyme. So angiotensinogen to angiotensin 1 with the help of renin, angiotensin 1 to angiotensin 2 with the help of angiotensin converting enzyme. Angiotensin 2 has variety of effects. That means it will cause the release of aldosterone from adrenal cortex. Number one, aldosterone from adrenal cortex. It will cause the release of that. Okay. And also, as the name indicates, angiotensin, it will cause vasoconstriction. And both of them, vasoconstriction as well as aldosterone, both of them can increase the blood pressure. It can increase the blood pressure directly by vasoconstriction. Vasoconstriction, as the name indicates, angiotensin, it can increase the tension inside the angio, inside the vessel, and also aldosterone. As a result of that, BP will increase. Now, how aldosterone can increase the BP? How aldosterone can ultimately cause changes in the BP? Try to understand that. My dear children, aldosterone can cause increased sodium reabsorption and increased potassium secretion. Ante blood gains dash and loses dash. Come on. These are the two A and B. A and B, blood gains dash and loses dash. This should be the result of this increased sodium reabsorption and increased potassium secretion. Come on. Yes, very good. I wrote part of the answer. Very good. So blood gains Na and loses potassium. But because Na is the chief cation of the extracellular fluid, this will increase. This will cause the increase in the osmolarity, increase in the blood osmolarity. Because, okay, they have lost K plus ion, equal number. But Na plus ion is the chief cation of ECF, extracellular. That means the plasma. Now, Na in plasma is more. As a result of that, there will be increased water reabsorption. And as a result of increased water reabsorption, increased blood volume. As a result of that, there will be increased blood pressure. So, you know that by decrease in the blood pressure, ultimately there is increase in the blood pressure. and aldosterone is causing the same. So aldosterone has actually caused the increased sodium and water reabsorption. Ultimately, the effect that can be seen in reabsorption is increase in the sodium and water reabsorption as the effect of aldosterone. Of course, there is increased potassium loss also, but that potassium loss, of course, there is there. But why I have written that yellow line? Because this is given directly in the NCERT. That is why whenever you find this, you can understand why this is happening. My dear children, the stimulus was decrease in the BP. The decrease in the BP, and as a result of that, directly increase in the BP by vasoconstriction, and indirectly increase in the BP by aldosterone. And this is how aldosterone will increase the BP. This mechanism is known as renin-angiotensin-aldosterone system. So please remember, I'm now going to tell you the golden statement of this entire last half an hour. Entire last half an hour, or let us say 20 minutes. Now the golden statement, remember, when the osmolarity of blood increase, whenever the osmolarity of blood increase, and whenever the blood pressure and also the GFR decrease. Either we can say blood pressure decrease or GFR, because both are together. Both will occur same. So whenever there is increase in the osmolarity, the osmoreceptors, particularly in the brain, they are stimulated. As a result of that, the pituitary will release ADH released from pituitary. Neurohypophysis. Hypophysis means pituitary. Neurohypophysis means posterior pituitary. Neurohypophysis. ADH is released. As a result of that, there is increased water permeability of collecting duct. As a result of that, more water reabsorption. As a result of that, urine concentration. Whenever the osmolarity of the blood increases. And why the osmolarity of blood increases? Osmolarity of blood increases whenever there is excess of fluid loss. Whenever the body loses water, whenever there is deficiency of fluid, osmolarity of the blood will increase. Whenever the blood pressure and GFR decrease, this will cause the activation of renin. So renin activation, this will cause the formation of angiotensin II, and as a result of that, the blood pressure increase. Okay, angiotensin II will cause increase in aldosterone. Aldosterone will cause sodium and water reabsorption. As a result of that, blood pressure will increase. So by both the ways, by aldosterone and also by directly working on the blood vessel, the blood pressure increase. Remember that these are the two ways, whatever given in the example, in the examination. For example, if they give you this thing, then your answer should be about ADH. And if they talk about this thing, then your answer should be about renin. It's an important board work that you see here, an important board work. These are two different questions, and these are the questions which will help us to gain that edge. I told you in the like in the beginning, questions that everyone do correctly. So of course we have to do them correctly, but we have to do the questions correctly which others are doing incorrectly. They are, fumbling in that. We have to become clear in that. This will give us an edge and we will be upper in the merit list, because it is not the passing of the examination or scoring 75% or distinction. Nothing like it is a competition. So other is running fast, you have to run faster than him. Simple, it's a competition. My dear children, let's talk about and how much of carbon dioxide is eliminated by the lungs. It is 180 liter. It is about 180 liter or 200 ml per minute CO2 elimination by lungs. Urea elimination by kidneys. Urea elimination by kidneys, how much? Yes, dash gram per day. How much? Dash gram per day. Dash means 25 grams per day. 25 grams per day of urea elimination by kidneys in the form of urine. In the form of urine. Urea elimination by kidneys in the form of urine. My dear children, so kidneys functions, these are the different kidney functions. And of course, kidney function is in eliminating the waste elimination, that then maintenance of blood pressure maintenance, then what? Then maintenance of pH of blood, okay? pH of blood maintenance. How much is the pH of blood? 7.4. Maintenance, very good. What else? Ionic concentration, the ionic concentration of blood maintenance, and not to forget the RBC production, increase in the RBC production. So erythropoietin release will increase erythropoiesis, erythropoiesis, POIESIS. And erythropoietin, this hormone is a protein hormone released by juxtaglomerular cells. Juxtaglomerular cells. These are the same. These cells, juxtaglomerular cells, the smooth muscles, modification of smooth muscles of afferent arteriole, they are doing that. Also, if you can remember, kidneys are also helping in activation of vitamin dash, activation of vitamin dash. Very important thing. This maintains the calcium It helps in calcium homeostasis, calcium balance, blood calcium balance, blood calcium balance. Come on. Blood calcium balance. Which hormone? Very good. Perfect, beta. D, vitamin D. Very, very good. Perfect. Vitamin D. Therefore, helping in calcium balance. That is also one of the functions. So, generally, we know this function. ठीक है, ये भी पता है, ये भी पता है. Okay. This given in the NCERT, given in the NCERT. Okay. Then, of course, this is also given. Erythropoietin and activation of vitamin D. In fact, last year, 2019, a question was there about the hemodialysis. Which of the following is not related to hemodialysis? And there is something about vitamin D or blood calcium balance. Blood calcium balance or vitamin D. I don't exactly remember that line, but I know that is based on this line, that activation of vitamin D. And when vitamin D is activated, there will be more absorption of more. Yes, it was absorption. Yes. It was absorption. I remember now. Absorption of calcium in gut. This was there. In the presence of vitamin D, calcium from the diet, from the food is absorbed. So this was there. Absorption of calcium in gut increases. I hope, guys. The muscle contraction, skeletal muscles type, skeletal muscle types. The type of body muscles already we know, smooth, skeletal, cardiac, but the skeletal muscles type are these two. We have got the white muscle fibers, white fibers and red muscle fibers, white and red muscle fibers. And which one we have? We have both of them. We have both of them. The difference is white and red muscle fibers is in their some biochemistry, some anatomy. Okay, so mainly we discuss the biochemistry differences. And remember, these are the skeletal muscle cells. So inside these white muscle fibers, we will find that the endoplasmic reticulum is more than the endoplasmic reticulum in the red muscle fibers. My dear children, the red muscle fibers, they have got more of mitochondria, so they have more of I wanted a thin pen. बच्चों, इनके पास more mitochondria. Here, mitochondria are lesser. Here, endoplasmic reticulum is limited. Here, endoplasmic reticulum is too concentrated. More endoplasmic reticulum, more mitochondria. This is the main difference you can see here. Do not get confused with the nuclei. Beta, ये तो multinucleated होते हैं. Don't worry about that. वो same है. They are same. They are brown nuclei. Green mitochondria. White endoplasmic reticulum. Then myoglobin, which is a muscle hemoglobin, is more here. Myoglobin, muscle hemoglobin, which is a oxygen storing pigment, is more in case of the red muscle fibers. And therefore they look reddish in color. They have got more mitochondria and more myoglobin. Here less mitochondria and less myoglobin, and of course the capillaries, the blood supply is also limited. And the type of work they do is of course different. Now they help in rapid contraction. They help in rapid contraction. After some time, there will be anaerobic respiration. Anaerobic respiration. This one is slow contraction, slow and sustained. Slow and sustained contraction. And they are mostly anaerobic respiration. Why? Because they have plenty of oxygen within them in the form of bound to myoglobin. Okay, they have plenty of mitochondria also, so that is helping in them, aerobic respiration. And in white it is aerobic respiration, and this is anaerobic respiration. Now, where these muscle fibers are used? See, whenever we want a slow and sustained contraction, for example, our back muscles, our posture muscles, flight muscles of kite, eagle, long time contraction, posture muscles, marathon runner, limb muscles in a marathon runner, you will find red muscle fibers. Now coming to this part, so what that was posture muscles, we can say eyelid muscles, the muscles that cause blinking of the eyes. Okay, then the flight muscles of sparrow, they are doing very fast contraction. Of course, whenever the fast contraction, the relaxation is also very fast, na beta? Relaxation is also very fast. Relaxation is also very rapid. Rapid contraction, rapid relaxation. Slow and sustained contraction. Slow and sustained. Flight muscles of a sparrow, it just hop from one point to another point. And also the limb muscles of a sprinter. Who is a sprinter? A sprinter is an athlete who runs short distances. A sprinter. He is an athlete who runs short distances. The shortest distance in the Olympics is 100 meters. The 100 meters, okay? So 100-meter runner, 200-meter runners, these are called sprinters. And the longer one, marathon, we have 800 meters, 400, 800, and more than 1 kilometer, they are marathon runners. Marathon, sprinter. Flat muscles of kite flies in the air for longer period. And they are aerobic fibers, they are anaerobic fibers. You can clearly see why they are aerobic. This is myoglobin, and myoglobin is a storing. It's a oxygen storage pigment. Hemoglobin is oxygen transport pigment. Hemoglobin is oxygen transport pigment. Myoglobin is oxygen storage pigment. My dear children, these are the types of skeletal muscles, and it is an important point to be noted in this chapter. Then in this chapter, joints are important. Joints. Teacher will give you questions on joints. In this chapter, if I tell you the last point about the muscles, that means how the calcium availability in the sarcoplasm increases whenever there is a nerve supply or a nerve impulse, it causes depolarization of the membrane, causes increase in the calcium ions, and this calcium ions, it binds to the troponin. As a result of that, the masking of the active site, so there is unmasking occurs, and therefore there is actomyosin formation. I repeat, beta, whenever a nerve impulse come to a muscle, a nerve impulse is just a wave of depolarization. It will cause depolarization of the muscle membrane also. As a result of that, calcium, because the calcium channels open up in the sarcoplasmic reticulum, and plasmic reticulum, these are actually the calcium storehouse. Calcium storehouse inside the sarcoplasm. Calcium availability increase. As a result of that, calcium bind to troponin. As a result of that, unmasking of the active site occurs, and actin and myosin combine, called actomyosin formation. And after that, there will be sliding of thin over the thick. Once again, sliding of thin over the thick. My dear children, annam, chutney, sambar, bhaji. I repeat, annam, chutney, sambar, bhaji. So ATP hydrolysis. For this, you have to open the diagram of NCERT. Cross bridge formation. Sliding or rotation, and breaking of cross bridge. Here, the main point that is asked is this breaking of cross bridge takes place by binding of ATP. Breaking of cross bridge takes place. At the time of power stroke, whenever there is a bending of the cross arm, whenever there is a power stroke, the ADP is released. From the active site. ADP plus inorganic phosphate IP, they are released. This breaking is only by binding, and this cross bridge formation is due to the energy obtained by hydrolysis. Last line, annam, chutney, sambar, bhaji. Okay. Energy obtained by hydrolysis, that energy causes cross bridge formation, and also the moment it forms, there is a power stroke. At the time of power stroke, ADP is released from the active site, and now new ATP binds, and this binding of ATP, it helps in the breaking or unlocking of the cross bridge, and the myosin head will again come back in its normal docking position. And again, a cycle will repeat. So in one cycle, at one head in one cycle, one ATP molecule is consumed. Okay? The type of joints in the body, some names are, for example, sutures. Sutures are immovable joints in the skull bone. immovable fibrous joints in the skull bone. Then symphysis. Another term is symphysis. These are the cartilaginous joints. These are the fibrous, white fibrous joints. They are totally immovable. And cartilaginous joints are quasi-movable, semi-movable. Example of symphysis is, yes, by the same name, symphysis pubis. Symphysis pubis. Between the two pubic bones. Sutures, example, between the skull bones. So these names are additional than the synovial joints. Synovial joints are the freely movable joints. So we have the ball and socket joint. Ball and socket joint is present in the shoulder as well as at the hip joint. Then we have the hinge joint. Which joint? Hinge. Present at the elbow and the knee, elbow, knee, and also between occipital bone and atlas and occipital bone, hinge joint. The atlas occipital joint is also called yes joint. Yes, because it causes nodding of the head, nodding, as if we are agreeing. We are saying yes, we are agreeing. My dear children, then we have other type of joint. For example, there is something called gliding joint. Gliding joint is a joint between carpals and tarsals. It's very little movement occurs, but no doubt about that, it is again a synovial joint. Next one, this joint is a saddle joint. And saddle joint examples are very limited. The example is first CM. Only remember first CM. First is first, CM stands for carpometacarpal. Carpometacarpal. My dear children, hope you understand all these, okay? So these are the types of joints. Among them, maximum range of movement occurs in the ball and socket joint. The ball and socket joint are formed between the head of humerus and glenoid cavity of scapula, head of humerus, glenoid cavity of scapula, and another one is between the head of femur and acetabulum of the coxal bone, head of femur, acetabulum of the coxal bone. They can also say that the joint between the head of humerus as well as in the pectoral girdle, correct? And joint between the head of femur with the pelvic girdle, that also correct. Okay, guys. Nervous system. In the nervous system, why the people are feeling nervous about the nervous system? Don't know why. Children, please understand this one. If I say time is here and here I'll take the voltage, transmembrane potential, transmembrane potential. A suggestion to all the children who are in the classroom as well as children who are online, if you have particular areas, you can say to me, if I find that that area is also an area where maximum more questions can be expected. For example, pedigree 50-50, but Hardy-Weinberg, not so likely from Hardy-Weinberg. Even though if you have Hardy-Weinberg problem, the people in the classroom, they can write on a piece of paper and they can send it to me. So after after some, just couple of chapters, I'll be taking your particular doubts. For example, the part B of the textbook, starting from reproductive system, in every chapter, 1 to 2 points, which are mainly the doubts, you can write it to me. I'll just see them, and if I find them that they are important, because this is not the doubt removal class. This is the place where I find many questions are expected. This is the discussion of that part. Of course, this will not be asked, but what is asked is this. This is minus 70 millivolt, minus 90 millivolt. 1 millivolt is 1 by 1000 of a volt. This is plus 40 millivolt, and this one is 0 millivolt. So minus 70 millivolt, minus 70 millivolt. Once the membrane is at minus 70 millivolt, what shall we call the membrane is? We'll call the membrane is polarized. Membrane is polarized. Polarized, what is the potential? Minus 70 millivolt. And a polarized membrane can be excited, particularly if the stimulus is of minimum threshold potential. Now, stimulus always causes sodium leakage. What the stimulus causes? Sodium leakage. Another way of saying, stimulus always causes sodium permeability increase in sodium permeability. But the magnitude is different. No? This much permeability, this much, that much. So the stimulus must be strong enough so that the sodium permeability should be strong enough to minimum cause a change of 20 millivolt. So minus 70 should come to minus 50 millivolt. So if it is lesser than that, it is not going to make any change. It will die soon. Even it dies soon. Okay. But as the change can cross this threshold, minus 50, then automatically more and more sodium permeability increase, more and more sodium permeability increase, and the potential can reach to the spike potential, which is plus 40. Now the sodium permeability totally stops, and then the potassium permeability increase. Potassium movement will occur from the high concentration to the low concentration. Do not forget, potassium is more inside the cell. So potassium will come out of the cell, and potassium will come out of the cell. That means plus is coming out of the cell. That means if one plus outside, one corresponding minus inside. You understand? So the cell is becoming minus from inside, negative from inside, negative from inside, negative from inside, negative from inside. It carries now this much of duration passed, it becomes negative. It becomes even more negative than the resting stage. Normal resting stage is polarized, it becomes even more negative. But then now the potassium permeability also stops. But then see, now the channel permeability stop, but the pump, exchange pump will pump out sodium and pump in potassium, pump out sodium, pump in potassium, pump out sodium, pump in potassium. My dear children, let us not get confused by this. This is just to show you. ऐसा ना लगे कि there are, okay. This is just to show you that if it is this much now, it is going to die. Only when it can cross the threshold, then it is able to cause the further increase in the sodium permeability so that it can cross zero and can become positive. Cell become positive from inside. How the cell become positive from inside? Because positive ion, sodium ion, enter inside the cell. Sodium ion enter inside the cell. I can call this as sodium influx inside, and this is potassium efflux, potassium out, sodium in, potassium out. When the channels open up, sodium in, potassium out. When sodium in, cell at what level become positive? It become positive up to plus 40 millivolt. We can say that if it was polarized here it is depolarized. It was polarized, it is now depolarized. We can say it is completely depolarized. And this is called depolarization. So this is called depolarization. So the depolarization by sodium influx. And similarly, Opposite is repolarization. And this is hyperpolarization. You can say this is hyperpolarized. Hyperpolarized. So normal polarized, stimulus depolarized, and again it is repolarized. But before becoming polarized, it becomes hyperpolarized. Tarvata it becomes polarized. From this point to this point, some time is required, and this period is known as, this period is called refractory period. Refractory means not excitable. It is not excitable. If you follow other things also, and if you do not follow other things, if you can answer these two questions, we will not trap you anymore. These are the questions that are asked. Yes, we can ask you that how come unidirectional flow of impulse takes place? In an axon, why can't the impulse return back? Because to return back, the back portion should be polarized. But when the impulse is in the reach, the previous portion is in the refractory period. By the time previous portion become polarized again, the impulse go beyond the reach. Okay, so that is the reason why unidirectional flow of impulse takes place. Also, this is also very important, very, very important, and this reminds me, this class only, and the coming days I will give you one question on this also. I have given question on reflexes. I'm happy with that. I'm going to give questions on this also, okay? So this is a chemical synapse. A chemical synapse, presynaptic knob, postsynaptic membrane, and the receptor for neurotransmitter is present on the postsynaptic membrane. The receptor for neurotransmitter present on the postsynaptic membrane, and the neurotransmitter is present inside these synaptic vesicles. Synaptic vesicles. So this is the situation, okay? And this is a dendrite. So this is a dendrite. And what is this? Axon terminal. Axon terminal. The nerve impulse reaches to the end, causing migration of these vesicles to the periphery. Which periphery? Ye ikada, this one. As a result of that, these vesicles will release the content in the synaptic cleft. Bacha, this is the synaptic cleft, the gap, cleft. Bacha, cleft, synaptic cleft, synaptic cleft. The neurotransmitter binds to the receptor. We have very, very easy way of asking this. We can ask you, where is the receptor? Very simple. If the receptor damages, then what? Of course, impulse will not be transmitted. What if the receptor binds to the neurotransmitter? What if? Then there is generation of a new potential in the dendrite. Which potential? Fixed potential? No. There is development of graded potential. Graded potential. The boys remember in the boys campus when we were discussing the question paper, I think day before yesterday, or just the last time when we discussed the paper, that time we have opened the PYQ book, and this graded potential was given in 2016 or maybe 2018 paper. So remember that one potential that you have known is the resting membrane potential. One potential known to you is depolarization potential, or better known as action potential. And these are the potential of varying degree. Varying degree. Therefore, we call them graded, until they are of various grades. They can also be positive or negative. They can also be excitatory or they can be inhibitory. They can be excitatory, they can be inhibitory, okay? And this is a dendrite actually. So in the dendrite, there is no action potential. Why? Because in the dendrite, the gated channel for sodium are not there. The sodium gated channels are absent here. Therefore, this is not going to give the same depolarization, because depolarization occurs by opening of the sodium gated channel. More sodium inside, more gated channel open, more the gates open, more sodium enter inside, more the gate open. I mean to say, because a doubt can come to your mind, because, sir, if these channels were open earlier, why can't the sodium enter earlier? Better, these are gated channels, channels with a gate. Normally gates are closed, but voltages or transmembrane potentials are known to cause changes in the gate. Also, if more sodium enter inside, this also causes opening. Very simple, as the cell is becoming positive from inside, more and more gated sodium gated channels open up. Please keep this point in mind. Wherein if it is an electrical synapse, it is in so close proximity. If it is an electrical synapse, it is in so close proximity that there is no such chemical, direct electrical signals are passed. They both look as if they are connected. Both the cells look as if they are connected. And yes, the cytoplasm is connected with the help of gap junctions. Cytoplasm is connected with the help of gap junctions. My dear children, in the nervous system, we have the central nervous system and peripheral nervous system. Peripheral nervous system. Central nervous system is very easy. We have brain and spinal cord that makes the central nervous system. Peripheral nervous system is with the help of the nerves. With the help of the nerves. Peripheral nervous system. Water break. Children, these nerves can be from the receptors to the CNS, or from the CNS to the effectors. The receptors can also be in the viscera or in the other body parts. Effectors can be muscles or glands. The muscles can be skeletal muscles or visceral. Okay. Skeletal muscles, all the signals coming to the skeletal muscles, those signals are carried by somatic nerves. Somatic nerves. And the signals to the visceral muscles, okay, these are carried by autonomic nerves, autonomic nerves. In fact, to the glands also, autonomic nerves. So what you find here, very important notice here, that autonomic nervous system comprise of only the efferent. Autonomic nervous system comprises of only the efferent fibers coming from CNS to the effectors, only the efferent fibers. Okay, autonomic nervous system. But when we say receptors, these receptors are on the body surface or in the viscera. Okay, so they can be in the surface of the body surface, general body, I should say, or they may be present in the viscera. Yes, we have. Can't you feel stomach pain? We feel. Can't we feel acidity? We feel. Receptors are there also. And from those receptor signals are carried. Okay. From those receptor signals are carried to the CNS. Okay. So, what shall we call the nerves which are carried from the visceral receptors to the CNS? The nerve carried from the visceral receptors, the nerve carried to the visceral receptors to the CNS, and also, okay, in the viscera, there are receptors and there are muscles or effectors. In the viscera, body viscera means soft organs of the body cavity. From the receptors, signals are going to the central nervous system. From the central nervous system, effectors are getting efferent. Some of them are also the reflexes. Some of them are reflexes. That means to the effectors, they are coming only because of the afferent. Only because of the afferent. This is afferent. This nervous system, what shall we call it? This is known as visceral nervous system. Visceral. Visceral nervous system is different from autonomic. How autonomic will look like? Autonomic will look like this. Autonomic is from the CNS to the effectors. That means the signals to the effectors are not due to the afferent. This efferent is not due to any afferent. They are not secondary. They are not result due to afferent. They do not result due to afferent signals. And the nervous system which carries the signals which are not due to afferent, this is a special one, and this we will call it as autonomic nervous system. This nervous system is autonomic nervous system, and this one will be called visceral nervous system. What is common in both of them? Common in both of them is that the effectors are in the viscera. What is common in both of them? Only thing common in both of them is the effectors are either the glands or the muscles. Effectors are either the glands or the muscles of the viscera. My dear children, this explains the chapter wherein the second page of this chapter, they have included the new prints after 2018, 2019. Those prints have included the term called visceral nervous system. And yeah, they have not given this point clearly, that they have not spoken about the visceral reflexes. There are many visceral reflexes which are operating. They have not spoken about that. Okay, so otherwise they have just mentioned afferent, efferent, that's it. Then you have to mug up or battico to the page of CNS. That means what the association neurons do, what all those, that is, you have to battico to. Okay, guys, and let's make a change, little bit change here. One very good hint I'll tell you. One very good hint. Mechanism of hormone action. Dear children, before understanding the mechanism, you have to understand there are two categories of hormones. They are water soluble and fat soluble. What are fat soluble hormones? Fat soluble hormones are two types. One are steroids and iodothyronine and iodothyronine. Neens, ante T3 and T4. Steroids are sex hormones, all the sex hormones of male and female, and aldosterone and cortisol. Aldo and corti. Aldosterone and cortisol from the adrenal cortex, only these are steroids. All the sex hormone, testosterone, estrogen, progesterone, even they can call it androgen, general term, all these are steroids. They are fat soluble. After this, rest all are water soluble. In mechanism of hormone action, just remember one thing, that the hormone which are water soluble, their receptors are membrane bound. They have membrane bound receptors. And the hormone which are fat soluble, they have intracellular or nuclear receptors Almost 90% questions are based on this. Where are the receptors? And if the receptor is membrane bound, there is a problem. How to communicate? If the receptor is membrane bound, the problem is, so there is binding of the hormone to the membrane bound receptor, and so ultimately you have to activate some enzymes. Okay, so here is the target to be acted upon. So how to communicate to this target? Maybe this is an enzyme to be activated. How to communicate to this target? They can communicate to this target with the help of second messengers. Who is the first messenger? First messenger. Second messengers are cyclic AMP, calcium ions, inositol triphosphate IP3, inositol phosphate, inositol phosphate, calcium, cyclic adenosine monophosphate. This can be activated or it can be inhibited, and this will give changes in the biochemical changes, and biochemical changes causes metabolic changes, means metabolic changes occur, and metabolic changes cause physiological changes. Physiological changes, very simple. They all are connected. The biochemistry depends on biocatalyst enzymes, and on biochemistry metabolism depends, and metabolism affects physiology. As a result, your first messenger has actually, this has caused changes in the physiology. Hormone action. The other hormone action is through the DNA, or we can say through the gene expression modification in gene expression. There is a receptor, a mobile receptor, this time a mobile receptor or a motile receptor. Hormone can cross the plasma membrane, so no need to have the membrane bound receptor, not required, and therefore second messenger not required. Hormone can bind to the free receptors, cytoplasmic or nuclear receptors, and can directly bind to the part of the genome and can affect gene modulation, can change gene, can affect transcription and translation. As a result of that, what will happen? As a result of that, enzymes are either activated means new enzyme formed, or enzymes are inhibited, means there is no formation of enzyme, and this affects the biochemistry, metabolism, and physiology. Same thing. That was doing directly working on the enzyme, and this is working on the DNA. These are steroids and T3, T4 iodothyronines. Human reproduction, menstrual cycle, average 28 days, but not fixed. Actually, we are preparing for 45 bits. What is in my mind is 45 bits. And 45 bits means a big syllabus. And now to 50 bits. 45 for you, but 50 for me. Ideally, one day should be for first year and one day should be for, one class should be for second year. Anyways, I'll try to Main point here, menstrual cycle, the phases are, these are the phases, नहीं गलत बन गया. Make cardiac cycle. Cardiac cycle. Cardiac cycle na atrial systole, ventricular systole, joint diastole. Here also, from first day to the fourth day, 5th to 14th day, 28th day. This is always fixed. Here comes first point. This duration is always fixed. Here comes first point. And this is of 14 days. This is of 14 days. Here comes first point. This is always fixed. It is of 14 days. This is 4 to 5 days. And the remaining part is this one. So what are this? Try to understand. This is known as the menstrual phase. Menstrual phase, that means the menstruation is going on, which includes the menstrual bleeding also, shedding of the endometrium, the inner membrane of the uterus, inner lining of the uterus, along with some blood, means menstrual phase. Then in this time, the hormones, the sex hormones are very, very less. Estrogen and progesterone are very, very less. As a result of that, it will cause GnRH pulse. GnRH pulse. Pulse means, why I'm using the word pulse? Because the right thing is pulse only. If you remember one bit came in PYQ, GnRH pulse frequency depends on, and the answer was circulating levels of estrogens and progesterone. GnRH pulse means GnRH is not poured like this, increased in the blood. GnRH comes in pulse. The pulse has different frequency. GnRH increase, decrease, increase, decrease. How many times this happens in one day? This is the frequency. And this causes the pulse frequency in such a way, GnRH pulse frequency, in such a way that this causes the release of follicle stimulating hormone. And what this hormone will do? Stimulate the follicle, na? It will stimulate the follicle. Follicle stimulating hormone will stimulate the follicle. As the follicle stimulates, what it will do? Come on. Which hormone will come out from the follicle? Which hormone comes out? And the level of the hormone increasing. Level is increasing. Yes, you are right. Estrogens. Estrogens. So estrogen, the bigger arrow means more and more estrogen released. Smaller size, some estrogen, bigger size, or bigger, more estrogen. And as the estrogen is so high, this causes the LH surge. LH surge. This is a positive feedback, means high estrogen causes another hormone high. Negative feedback is one hormone high, other hormone low. Negative. But if both are high or both are low, positive. This is a positive feedback. LH surge. This will cause breaking of this. LH is luteinizing hormone. Ovulation will occur and rest of the cells become corpus luteum, and hence the name luteinizing. Rest of the cells become corpus luteum, hence the name luteinizing hormone. And LH is maintaining the luteinizing hormone. LH is maintaining the corpus luteum. LH. Guys, never ever forget that this is the source of another hormone, another female sex hormone by the name progesterone. Progesterone chinna, progesterone equa. Why? size pedda, big size, more progesterone. Along with progesterone, it also produces estrogens. So there will be another peak of estrogen coming soon. This high level of estrogen and progesterone is not liked by the hypothalamus. Hypothalamus changes the GnRH pulse frequency. As a result of that, LH is withdrawn. LH start decreasing. And LH decrease to kya hoga? Corpus luteum will start shrinking. Corpus luteum dies as a white scar. Corpus luteum dies. And corpus luteum dies, so kya hoga? What will happen? Corpus luteum is the source of estrogen and progesterone. So in this period, from here to here, the estrogen and progesterone, they start decreasing. As the progesterone is very, very low, it will cause the menstruation. As the progesterone is very low, it will cause menstruation. This menstruation, the reason for this menstruation is very, very low progesterone. Very, very low progesterone. And again, the reason for this FSH release, who will tell me? What is the reason for this change in the GnRH pulse frequency? Yes, low ENP. Because hypothalamus is not anti to any hormone. Hypothalamus wants to maintain a balance. If E and P, E means estrogen, P means progesterone. If they are increasing, hypothalamus will stop them. And hypothalamus has got the key. Hypothalamus can control the pituitary. And pituitary gonadotropins are controlling the gonads. Gonadotropin, gonads. Ovary, these are the gonads. Gonadotropin, LH, FSH. My dear children, what is to be noted down here is that this menstruation is due to very low progesterone. This release of FSH is also due to very low progesterone. That means progesterone become low here and low progesterone maintained in this period. And this is from here itself, it causes changes in the frequency and again follicle increase occurs. This can be seen again by the graph given in the NCERT. Which hormone attains double peak? Which hormone attains double peak? Okay, first of all, which hormone attain peak? Some hormones attain peak. Try to recall from the diagram. Some hormones attain peak. In the online one, yesterday I have shared the NCERT diagrams. So that is a very good way of revising the main points of NCERT. Just keep on seeing the diagrams, and particularly the time when you are having some other work, you are, I can tell myself, I can use lot of reading takes place in bathroom. Okay, so that time can be utilized. Okay, and so we can revise the things from the NCERT diagram. We have LH, FSH, estrogens, and progesterone. These hormones attain peak in the menstrual cycle. Now, if I ask you in this phase, in the follicular phase, which hormone attain peak in the follicular phase? LH and FSH attain. Actually, LH is just here, but we can say both of them attain peak in the follicular phase. Along with that, estrogen attain peak in the follicular phase. This is called luteal phase. Luteal. Which hormone attain peak in the luteal phase? Answer is estrogen and progesterone attain peak in the luteal phase. both estrogen and progesterone. That means estrogen attains peak twice. Estrogen attains peak twice. This is the hormone which attains peak twice. I know time is over. Actually, time is not over. I know, but you guys are hungry. And in hunger, see, I'm excited, so maybe the hunger is not felt, but you guys are hungry, and that's true. And it is better to eat the food at the right time, not to delay it. Okay, guys? Very good. Pedigree? Pedigree online, na? Pedigree online. Offline. Okay. What we can do, just remind me better. Whenever we have time, we can just announce that. And those who want to join, maybe 10, 15 children, those who want to join, and online and offline both, then we will do. Like today, I brought the samples of pedigree. And let me just check and talk to the children here, whether they want after the lunch break or not. Let us see. But you have a test now. All the very best for the test. And we will be doing all the things, later on. Bye-bye. Take care. Band karo. Bye-bye. God bless you, children. God bless you. God bless you.